Hello there. I'm so glad you found your way here tonight. Right now, as you settle in, imagine a creature so small it fits between grains of sand. A creature so ancient it has walked the earth for over 500 million years. A creature so resilient that it can survive the vacuum of space, the crushing depths of the ocean, temperatures near absolute zero, and radiation that would destroy almost any other form of life. This is the tardigrade, also known as the water bear or moss piglet, a microscopic wanderer that looks like a tiny, chubby bear with 8 stubby legs and a slow, deliberate gait. Tonight, we journey into the hidden world of these extraordinary survivors. We will explore over 100 soothing facts about tardigrades, from their ancient origins to their remarkable ability to pause life itself, from their presence in your garden moss to their accidental journey to the moon. There is no test at the end, no rush, no urgency, just the gentle unfolding of one of nature's most astonishing survival stories to carry you softly towards sleep. So let your breathing slow, let your thoughts grow quiet, and allow yourself to drift into the peaceful wonder of the microscopic world. Welcome to Drowsy Discovery. I am glad you found your way here tonight. Perhaps you have had a long day. Perhaps you simply enjoy falling asleep to thoughts about the remarkable creatures that share our world. Either way, this space is for you. There is something deeply calming about contemplating the quiet resilience of tardigrades. Every slow step across a moss leaf, every transformation into a dormant ton, every revival after decades of stillness carries within it a story millions of years in the making. You are connected to this story more than you might realize. The same water that flows through your body also sustains these tiny wanderers. The same atoms that form your cells were forged in the same ancient stars that scattered matter across the cosmos billions of years ago. If you enjoy these gentle journeys, I invite you to like, subscribe, or share a thought below. It helps others find their way here too, one sleepy soul at a time. But for now, all you need to do is relax. So settle in, let the weight of the day drift away, and let us begin with the first of many quiet truths about tardigrades. The tardigrade is patient beyond measure. It can wait out droughts that last decades. It can endure temperatures that would destroy most living things. It can curl into a tiny ton and let years pass without noticing. And yet, within all that stillness, there is an ancient resilience, a survival strategy perfected across 5 mass extinctions, a biology so unusual that scientists are only now beginning to understand its secrets. Tonight, we will wander through this magnificent microscopic world. We will meet the creatures that outlasted the dinosaurs. We will discover the strange proteins that shield their DNA from harm. And we will uncover truths about tardigrades that may surprise even the most curious minds. But there is no hurry. Tardigrades have been waiting for hundreds of millions of years. They can wait a little longer while you rest. Let us begin with what makes these tiny creatures so extraordinary. Tardigrades can survive in the vacuum of space. In the cold emptiness beyond our atmosphere, where there is no air pressure, no oxygen, and temperatures swing between searing heat and freezing cold, most living things would perish within seconds. Tardigrades, however, have proven that even this hostile environment cannot defeat them. When exposed to the vacuum of space, these microscopic creatures enter a state of suspended animation, drawing their legs inward and forming a compact structure known as a ton. In this dried, dormant form, their cells are protected by special proteins and sugars that replace the water normally surrounding delicate biological structures. Without water to freeze or boil, and with their metabolism reduced to virtually nothing, they can endure the vacuum for days at a time. Space agencies have confirmed this by placing tardigrades on the exterior of spacecraft in low Earth orbit, and many survived the ordeal and revived when returned to water. Their ability to withstand conditions that are lethal to nearly every other form of life makes them one of the most resilient creatures ever studied. They withstand temperatures hotter than boiling water. Most organisms begin to suffer severe damage when temperatures climb above roughly 40 degrees Celsius, as the proteins that drive cellular processes start to unfold and lose their shape. Tardigrades, in their remarkable ton state, can endure temperatures well above the boiling point of water, surviving brief exposure to around 150 degrees Celsius in laboratory conditions. At such temperatures, ordinary proteins would be destroyed and cell membranes would disintegrate, yet tardigrades persist. The secret lies in the way they prepare for extreme heat. As they dehydrate and enter dormancy, special molecules form a glassy matrix around their cells, locking delicate structures in place even as the surrounding temperature soars. This vitrified state acts like a biological shield, preventing the kind of molecular chaos that would tear apart an unprotected cell. When the heat passes and water returns, the glass softens, cells rehydrate, and the tardigrade slowly returns to life. It is a quietly astonishing feat of survival engineering, perfected over hundreds of millions of years of evolution. They endure cold, approaching absolute zero. Absolute zero, at roughly -273 degrees Celsius, is the lowest temperature theoretically possible, the point at which all molecular motion would cease. Tardigrades have been shown to survive exposure to temperatures only a fraction of a degree above this extreme limit. At such cold, the water inside most cells would form sharp ice crystals that pierce and destroy delicate membranes. Tardigrades avoid this fate by entering their dried ton state before the cold arrives, replacing their internal water with protective sugars and proteins that prevent ice formation altogether. In this glassy, vitrified condition, their cells are effectively frozen in time without the damage that ice would cause. Scientists have immersed tardigrades in liquid helium and liquid nitrogen during experiments, and a significant proportion revived once gently warmed and rehydrated. The fact that a living creature can brush so close to the absolute floor of temperature and still wake up is a testament to the extraordinary chemistry that evolution has built into these tiny bodies. They survive complete dehydration. Water is considered one of the absolute essentials of life. Every known organism depends on it for cellular processes, chemical reactions, and the maintenance of biological structures. Yet tardigrades can survive losing almost all of their body water, drying out to a degree that would be instantly fatal to virtually any other animal. When their environment dries up, tardigrades gradually pull in their legs, contract their bodies, and form a compact ton in which water content can drop below 3%. In this state, their cells do not simply collapse. Instead, special proteins and sugars step in to take the place of water, supporting membranes and stabilizing molecules that would otherwise crumble. The tardigrade essentially pauses its own biology, existing in a state that is neither fully alive nor truly dead. When moisture returns, water seeps back into the ton, cells rehydrate, and the tardigrade unfolds its legs and resumes its quiet life as though nothing happened. This process, called anhydrobiosis, is one of the most remarkable survival strategies in the natural world. They shrink into a suspended state called the ton. When conditions become hostile, whether through drying, freezing, or exposure to toxic substances, tardigrades undergo a transformation that is both simple and extraordinary. They retract their 8 legs, expel most of their internal water, and curl into a tiny barrel-shaped structure called a ton. In this form, the tardigrade is scarcely recognizable, shrinking to a fraction of its normal size and appearing almost like a speck of dust. Yet within that tiny package, the essential machinery of life is preserved with remarkable precision. The tun state is not death but a profound pause, a suspension of virtually all metabolic activity that allows the creature to wait out conditions that would destroy it in its active form. Heart rate, breathing, and cellular processes drop to levels so low they are almost undetectable. The tun can be carried by wind, washed by rain, or frozen for years, and the tardigrade inside remains viable. When favorable conditions return, the tun absorbs water, the body swells, the legs extend, and the animal resumes its slow walk across the moss. Some have revived after decades without water. The patience of a tardigrade in its tun state is almost beyond comprehension. There are documented cases of tardigrades reviving after spending decades in a completely desiccated condition, stored in museum collections or dried moss samples that were set aside and forgotten. In one well-known case, tardigrades recovered from a moss sample that had been kept dry for over 30 years, returning to life within hours of being rehydrated. During all those years, the tardigrades existed in a state where metabolism was effectively zero, with no food, no water, and no detectable biological activity. Yet the protective chemistry within their cells held firm, preserving membranes, proteins, and DNA against the slow degradation that time inevitably brings. When water finally returned, the molecular machinery started up again as though only moments had passed. This ability to bridge decades of time in suspended animation makes tardigrades one of the most patient organisms on Earth. It is a quiet kind of endurance that asks for nothing more than a drop of water to begin again. They resist radiation that would kill most forms of life. Radiation is one of the most destructive forces a living cell can encounter. High-energy particles and rays tear through DNA, breaking the delicate molecular chains that carry genetic instructions. For most organisms, even moderate doses of radiation are lethal, causing irreparable damage to cells. Tardigrades, however, can withstand radiation doses hundreds of times greater than what would kill a human being. Their resilience comes partly from a unique protein called DSUP, short for damage suppressor, which binds to DNA and physically shields it from radiation-induced harm. In addition, tardigrades possess highly efficient DNA repair mechanisms that can stitch broken strands back together after exposure. When in their dried tun state, the tightly packed configuration of their genetic material further reduces the target area for radiation damage. Scientists have exposed tardigrades to gamma rays, ultraviolet light, and heavy ion radiation, and significant numbers have survived and resumed normal activity. This extraordinary resistance has drawn the attention of researchers hoping to learn how tardigrade proteins might one day protect human cells. They endure pressures greater than the deepest ocean trenches. The deepest point in our oceans, the Challenger Deep in the Mariana Trench, sits under a crushing pressure of roughly 1,000 atmospheres. Very few organisms can survive at such depths, where the weight of the water above compresses everything with immense force. Tardigrades, remarkably, have been shown to endure pressures far exceeding even this extreme, surviving in laboratory experiments at pressures of several thousand atmospheres. At such levels, the molecular structures of most living cells would be crushed and distorted beyond repair. Tardigrades in their tun state, however, resist this compression because their cells are already in a dehydrated, vitrified condition with very little free water to be squeezed. The glassy matrix that protects their internal structures holds firm under pressure, much like a solid object, resists being crushed more effectively than a water-filled balloon. When pressure is released and the tardigrades are gently rehydrated, they return to normal activity. This tolerance suggests that tardigrades could potentially survive in environments far more extreme than anything found on Earth. They survive exposure to harsh industrial chemicals. Beyond physical extremes of temperature, pressure, and radiation, tardigrades have also demonstrated a surprising tolerance for chemical assaults. In laboratory settings, researchers have exposed tardigrades to various harsh substances, including organic solvents and other industrial chemicals that would dissolve or destroy most biological tissues. While active tardigrades are more vulnerable, those in the dried tun state show remarkable resistance because their cells are encased in a protective glassy matrix with virtually no free water for chemicals to interact with. The absence of active metabolism means there are fewer ongoing reactions that toxic substances could disrupt. In effect, the tun acts like a chemically inert capsule, sealing the tardigrade's biology away from the outside world. This does not mean tardigrades are invincible, as prolonged or extreme chemical exposure will eventually overwhelm their defenses, but their ability to shrug off brief encounters with substances that would be immediately lethal to most organisms adds yet another layer to their already extraordinary survival repertoire. It is one more quiet testament to the power of their dormancy strategy. They survive shock waves that mimic meteor impacts. Scientists curious about the limits of tardigrade resilience have subjected them to shock waves that replicate the violent forces of meteor impacts. In these experiments, frozen tardigrades were fired at high velocities into sand targets, simulating the kind of impact that would occur if a rock-bearing microscopic life crashed into a planetary surface. At lower impact speeds, a proportion of the tardigrades survived, suggesting that under certain conditions, they could theoretically endure the violence of an interplanetary collision. This finding is significant because it supports the idea that microscopic life could potentially travel between worlds aboard fragments of rock blasted into space by asteroid strikes. If tardigrades can survive such impacts, then the transfer of life between planets, a concept known as lithopanspermia, becomes at least theoretically possible. At higher velocities, however, the forces exceeded even tardigrade tolerance, reminding us that there are limits to their endurance. Still, the fact that any multicellular animal can survive even modest impact shocks is a remarkable testament to the toughness hidden within their tiny bodies. Their lineage reaches back over 500 million years. Tardigrades are not newcomers to our planet. Their evolutionary lineage stretches back over 500 million years, placing their origins deep in the Cambrian period, when the first complex animal life was beginning to diversify in the ancient oceans. At that time, the continents were arranged in configurations we would not recognize. The atmosphere had a very different composition, and the land was barren and lifeless. Somewhere in those primordial seas, the ancestors of modern tardigrades were already taking their first slow steps. Over the vast span of time that followed, continents drifted, oceans formed and disappeared, and entire ecosystems rose and fell, yet tardigrades endured. Their body plan has proven so successful that it has persisted through geological upheavals that wiped out countless other lineages. The fact that these tiny creatures were alive before the first fish swam, before the first plants colonized land, and before the first dinosaurs hatched is a humbling reminder of the depth of evolutionary time. Tardigrades are living relics of an ancient world, quietly carrying their history with them into the present. They outlived all 5 major mass extinctions. Over the course of Earth's history, 5 catastrophic mass extinctions have swept through the biosphere, each one wiping out a significant proportion of all living species. The most devastating of these, The end Permian extinction, roughly 250 million years ago, killed an estimated 90% of all species on the planet. Yet through every one of these cataclysms, tardigrades survived. Their ability to enter a dormant ton state, tolerating extremes of temperature, pressure, radiation, and drought, gave them a survival advantage that few other organisms could match. While giant reptiles, vast forests, and entire marine ecosystems were obliterated, tiny tardigrades curled into their tons and waited for conditions to improve. When the dust settled and the world slowly recovered, tardigrades were still there, ready to resume their quiet existence. This record of survival across 5 global catastrophes is unmatched by almost any other animal group. It speaks to a resilience that is not about strength or speed, but about patience and the ability to simply endure when all else falls away. Fossilized tardigrade remains are almost never found. Despite their extraordinary antiquity, tardigrades have left almost no trace in the fossil record. The reason is simple but frustrating for paleontologists. Tardigrades are tiny, soft-bodied creatures with no hard shells, bones, or teeth to preserve. Their bodies are made almost entirely of soft tissue encased in a thin, flexible cuticle that decays quickly after death. For a fossil to form, an organism typically needs hard parts that resist decomposition, or it must be buried rapidly under exceptional conditions that prevent decay. Tardigrades rarely meet either criterion. The sediments in which they live are often disturbed, and their microscopic size means they are easily overlooked, even when conditions for preservation are favorable. As a result, the fossil record of tardigrades is almost entirely blank, consisting of only a handful of specimens preserved in amber or other exceptional deposits. This scarcity makes it difficult for scientists to trace the precise evolutionary history of tardigrades through geological time. Much of what we know about their deep past must be inferred from genetic comparisons with living relatives rather than from direct fossil evidence. The first confirmed fossil tardigrade was only discovered in 2021. For decades, the tardigrade fossil record consisted of just a few ambiguous specimens, none of which provided clear anatomical detail. That changed dramatically when in 2021, researchers announced the discovery of a beautifully preserved tardigrade encased in a piece of Dominican amber dating back roughly 16 million years. The specimen was exquisitely detailed, revealing the animal's claws, mouthparts, and even the fine texture of its cuticle. It was a sign to a new genus, providing the first unambiguous window into the ancient anatomy of tardigrades. The amber had acted as a perfect preserving medium, trapping the tiny creature in a translucent golden tomb that protected it from decay for millions of years. This discovery was celebrated not only for its scientific value, but also for the sheer improbability of finding such a delicate animal preserved so well. It confirmed that tardigrades have looked remarkably similar to their modern descendants for at least 16 million years, suggesting that their body plan is both ancient and deeply stable. The find also hinted that many more tardigrade fossils might be hiding in amber collections around the world, waiting to be noticed. Their ancestors first crawled through prehistoric seas. Long before tardigrades colonized the mosses and lichens of dry land, their earliest ancestors were marine creatures, crawling slowly across the floors of ancient seas. The Cambrian oceans, teeming with the first explosion of complex animal life, would have been their original home. In those warm, shallow waters, early tardigrades likely fed on algae and bacteria, using primitive mouthparts to pierce cells and extract nutrients. The marine environment provided a relatively stable setting in which their basic body plan, 8 legs, a tubular body, and a simple nervous system could develop and refine over millions of years. Some tardigrade species still live in the ocean today, a living reminder of this aquatic heritage. Over vast stretches of time, certain lineages gradually adapted to freshwater habitats and eventually to the thin films of water clinging to mosses and soil on dry land. Each step in this transition required new survival strategies, including the ability to tolerate drying, which would eventually become the tardigrade's most famous skill. Their journey from sea to land is a quiet epic of adaptation written across hundreds of millions of years. They were among the earliest animals to colonize moist land habitats. The transition from water to land is one of the great dramas of evolutionary history, and tardigrades were among the earliest animals to make that journey. While larger creatures required millions of years to develop lungs, limbs, and waterproof skins, tardigrades found a simpler solution. They colonized the thin films of water that cling to mosses, lichens, and soil particles, environments that are technically on land, but still provide the moisture their bodies need. In these damp microhabitats, tardigrades could breathe by absorbing oxygen directly through their skin, feed on plant cells and microorganisms, and reproduce without ever needing to venture into open water. Their small size was an advantage here, because a droplet of dew or a thin layer of rainwater was enough to sustain their entire world. When conditions dried out, they could simply enter their ton state and wait. This strategy allowed tardigrades to establish themselves in terrestrial environments long before many other animal groups had begun their own transition to land. They were quiet pioneers of a world that was still largely empty and waiting to be colonized. Their exact position on the tree of life is still debated. Despite being studied for over 2 centuries, tardigrades still occupy an uncertain position on the tree of life. Scientists agree that they belong to the broader group of animals known as ecdysozoans, creatures that grow by shedding their outer skin through a process called molting. This places them in the same grand branch as insects, spiders, crustaceans, and nematode worms. However, the precise relationships within that group remain a matter of ongoing debate. Some genetic studies suggest tardigrades are most closely related to arthropods, the vast group that includes insects and crustaceans. Others place them closer to nematode worms based on certain molecular similarities. Still others propose that tardigrades sit on their own unique branch, separate from both arthropods and nematodes. The difficulty arises partly from their extreme antiquity and partly from the horizontal gene transfer events that have introduced foreign DNA into their genomes. Each new study seems to shift the picture slightly, and a definitive answer remains elusive. For now, tardigrades occupy a fascinating gray zone in evolutionary biology, a group whose deep relationships continue to puzzle even the most determined researchers. They share deep evolutionary roots with insects and velvet worms. Among the many hypotheses about tardigrade relationships, one of the most compelling places them close to 2 other ancient groups, the arthropods, which include insects, and the onychophorans, commonly known as velvet worms. All 3 groups share body plans built on repeated segments and paired limbs, and all grow by periodically molting their outer covering. Velvet worms, with their soft, caterpillar-like bodies and stubby, unjointed legs, are often described as a missing link between worms and arthropods, and tardigrades share certain features with both. The lobed legs of tardigrades resemble those of velvet worms, while their clawed feet and molting cuticle are more arthropod-like. Genetic analyses have sometimes grouped all 3 together in a clade called Panarthropoda, suggesting they descended from a common ancestor deep in the Precambrian. If this grouping is correct, then the ancestor of tardigrades, insects, and velvet worms was alive before the Cambrian explosion, making their shared lineage one of the oldest in the animal kingdom. It is a remarkable thought that the tiny water bear on a moss leaf is a distant cousin of the butterfly and the beetle. They show signs of horizontal gene transfer from other organisms. One of the most surprising discoveries about tardigrade biology is that their genomes contain genes that appear to have been acquired from entirely different organisms through a process called horizontal gene transfer. Unlike the usual inheritance of genes from parent to offspring, horizontal gene transfer involves the movement of genetic material between unrelated species, a phenomenon more commonly associated with bacteria than with animals. In tardigrades, researchers have found genes that seem to originate from bacteria, fungi, plants, and archaea, incorporated directly into the tardigrade genome. The exact mechanism by which this happens is still debated, but one hypothesis suggests that when tardigrades rehydrate after long periods in their tun state, their cell membranes are temporarily porous, allowing foreign DNA to enter and occasionally integrate into their chromosomes. These borrowed genes may contribute to some of the tardigrade's extraordinary survival abilities, providing enzymatic tools for repairing damage or tolerating extreme conditions. The proportion of foreign genes varies between species and studies, but the phenomenon itself is now widely accepted. It adds yet another layer of intrigue to an animal that never ceases to surprise. Their evolution includes unique survival proteins found nowhere else. Tardigrades have evolved a suite of survival proteins that are entirely unique to their phylum, found in no other known group of organisms. The most famous of these is DSUP, or damage suppressor protein, which physically wraps around DNA and shields it from radiation and other damaging agents. But DSUP is only one member of a broader family of intrinsically disordered proteins that tardigrades produce in response to desiccation and stress. These proteins lack the rigid three-dimensional structure typical of most biological molecules. Instead, they are flexible and shapeless in normal conditions, only forming protective structures when the cell begins to dry out. As water leaves the cell, these disordered proteins assemble into a glassy matrix that holds membranes and other molecules in place, preventing the collapse that would normally accompany dehydration. When water returns, the proteins dissolve back into their shapeless state, releasing the structures they were protecting. This elegant on-demand protection system is a product of hundreds of millions of years of evolutionary refinement. It represents a solution to the problem of survival that no other branch of the tree of life has independently discovered. Most tardigrades are smaller than a grain of sand. The typical tardigrade measures between 1/10 and 1/2 millimeter in length, making it far too small to see with the unaided eye. To put that in perspective, a single grain of fine sand is roughly the same size as the largest tardigrade species, and most are considerably smaller. You could place dozens of them side by side across the width of a fingernail and still have room to spare. Despite their tiny dimensions, tardigrades possess a complete and surprisingly complex body plan, with a brain, a nervous system, muscles, a digestive tract, and reproductive organs all packed into a body smaller than a full stop on a printed page. Under a microscope, they come alive as plump, translucent creatures with 4 pairs of stubby legs ending in tiny claws. Their movements are slow and deliberate, each step a careful placement that gives them their distinctive lumbering gait. It is easy to forget that these animals are all around us, living in the moss on rooftops, the soil in gardens, and the sediment at the bottom of ponds. Their invisibility to the naked eye is part of what makes them so quietly remarkable. Some curl into tight balls when disturbed. When a tardigrade senses danger, whether from drying conditions, sudden temperature changes, or physical disturbance, one of its first responses is to curl into a tight ball. This defensive posture is a precursor to the full-ton state, a rapid withdrawal of the legs and contraction of the body that reduces the animal's exposed surface area. By curling inward, the tardigrade protects its softer ventral side and tucks its vulnerable legs away from harm. This behavior has been observed in active tardigrades that are prodded under the microscope or exposed to sudden changes in their environment. It is a reflex that buys the animal time, allowing it to assess the threat before committing to the more energy-intensive process of forming a complete ton. In some species, the curling response is so swift that the tardigrade can go from active walking to a defensive ball in a matter of seconds. It is a small but telling detail about tardigrade biology, revealing that even these famously tough creatures prefer to avoid trouble when they can. Their first instinct is not to fight, but to withdraw, a strategy that has served them well for hundreds of millions of years. They walk on 4 pairs of stout, jointed legs. One of the most endearing features of tardigrades is their mode of locomotion. They walk on 4 pairs of short, stout legs, 8 in total, each one jointed and tipped with tiny claws or adhesive pads depending on the species. Their gait is slow and methodical, with each leg moving in a coordinated pattern that gives them a rolling, bear-like stride. Under the microscope, watching a tardigrade walk is surprisingly captivating. The legs extend and grip the substrate, pulling the plump body forward in an unhurried procession that can seem almost comical given the creature's stubby proportions. Despite their lumbering appearance, tardigrades are remarkably sure-footed, able to navigate the complex 3-dimensional landscape of moss filaments, lichen crusts, and soil grains with quiet confidence. Their legs are powered by individual sets of muscles, and the claws at the tips allow them to grip uneven surfaces securely. The 4 pairs of legs are arranged symmetrically along the body, giving the tardigrade a bilaterally symmetrical appearance that is one of the features linking them to the broader arthropod group. Every slow step is a small act of ancient engineering. Each foot ends in tiny hooked claws. At the tip of each of a tardigrade's 8 legs sits a set of tiny claws, often hooked or branched, that allow the animal to grip the surfaces it walks across. These claws vary in shape and number between species and are one of the key features used by taxonomists to identify and classify different tardigrade groups. Some species have simple, curved claws that hook into moss fibers and soil particles, while others have double claws or forked tips that provide a more secure grip on smooth or irregular surfaces. Under high magnification, these claws are exquisitely detailed, looking like miniature grappling hooks scaled to a world measured in micrometers. The claws are made of a tough material similar to the cuticle that covers the rest of the body, and they are replaced each time the tardigrade molts. For an animal that spends its life navigating the tangled architecture of mosses, lichens, and sediment, having reliable grip is essential. Without their claws, tardigrades would be unable to anchor themselves against the flow of water or the pull of gravity in their tiny but complex habitats. These small structures are a perfect example of how even microscopic creatures are finely adapted to their world. Their slow lumbering gait inspired the name water bear. The common name water bear was inspired by the way tardigrades walk. Under the microscope, their plump bodies and slow, deliberate stride bear a striking resemblance to a miniature bear lumbering across a landscape. The comparison is surprisingly apt. Just as a bear places each paw carefully on the ground, a tardigrade extends and retracts its 8 stubby legs in a steady, rhythmic pattern that looks almost comically ponderous given its microscopic size. The name has stuck for centuries, capturing both the appearance and the temperament of these tiny animals in 2 simple words. Early naturalists who first observed tardigrades through primitive microscopes were charmed by this bear-like quality, and the nickname quickly became a part of the popular vocabulary surrounding these creatures. In some languages, the common name translates similarly, always evoking the image of a small, round, slow-moving animal going about its business with quiet determination. There is something deeply appealing about the idea that among the smallest animals on Earth, one should walk with the unhurried confidence of a bear. It is a detail that endears tardigrades to everyone who encounters them. They are also affectionately known as moss piglets. Beyond the name water bear, tardigrades have another charming nickname, moss piglets. This name reflects their habitat as much as their appearance, evoking the image of a tiny, plump creature rooting around in the damp cushions of moss like a piglet in soft earth. Mosses are one of the most common habitats for tardigrades, providing the thin films of water they need to remain active and the microorganisms they feed on. The term moss piglet has a gentle, affectionate quality that captures the endearing nature of these animals. Under the microscope, certain species do look remarkably like miniature pigs, with rounded bodies, short snouts, and stubby legs that give them a rotund, well-fed appearance. Both nicknames, water bear and moss piglet, reflect the same basic observation, that tardigrades, despite being microscopic invertebrates, have an oddly mammalian charm about them. This has no doubt contributed to their popularity in recent years, as images and videos of tardigrades have spread across the internet, turning them into unlikely ambassadors for the microscopic world. Their appeal lies not in fearsome size or dramatic behavior, but in their quiet, unassuming, and oddly adorable presence. Many species have completely transparent bodies. One of the most visually striking features of many tardigrade species is their transparency. Under a microscope, the bodies of these tiny creatures can appear almost entirely see-through, allowing an observer to watch the internal organs at work in real time. The gut, the muscles, the reproductive system, and even the movement of food through the digestive tract can all be seen through the thin, translucent cuticle that forms their outer covering. This transparency is not universal among tardigrades. Some species are pigmented, displaying colors that range from pale yellow to deep orange or even red, depending on their diet and habitat. But many of the species most commonly encountered in moss and lichen samples are beautifully clear, like tiny living windows into the workings of a microscopic body. For scientists, this transparency is a tremendous advantage. It allows researchers to study the internal anatomy and physiology of tardigrades without dissection, simply by placing them under a microscope and observing. For anyone who has the chance to see a living tardigrade through a lens, the experience is unforgettable, a glimpse into a hidden world of quiet complexity. Their guts can glow under certain microscope conditions. When observed under certain types of microscope illumination, such as polarized light or fluorescence, the digestive tract of a tardigrade can appear to glow with an almost ethereal light. This effect is not true bioluminescence, but rather the result of how light interacts with the contents and lining of the gut. The food within a tardigrade's digestive system, often algal cells rich in chlorophyll, can fluoresce green under ultraviolet light, creating a vivid internal glow that traces the path of the gut through the transparent body. In some cases, the gut lining itself produces a faint fluorescence due to natural pigments or proteins. This visual effect has made tardigrades a favorite subject for microscopy enthusiasts who share vivid images of glowing tardigrade interiors online and in scientific publications. It is a beautiful demonstration of how microscopy can reveal hidden colors and patterns invisible to the naked eye. The glow serves no known biological function for the tardigrade itself, but it gives us a stunning visual window into the tiny creature's biology. It is one of those quiet surprises that reward anyone patient enough to look closely at the microscopic world. Simple eye spots allow them to sense light and dark. Tardigrades do not have complex eyes with lenses and retinas like those of vertebrates or even many insects. Instead, most species possess simple eye spots, tiny clusters of light-sensitive cells located on either side of the head. These eye spots cannot form images or detect shapes, but they can distinguish between light and dark, allowing the tardigrade to orient itself in its environment. For a creature that lives in the thin films of water on moss and lichen, knowing which direction light is coming from is useful. Many tardigrade species are negatively phototactic, meaning they tend to move away from bright light and towards darker, damper areas where conditions are more favorable for survival. The eye spots themselves are often visible under a microscope as tiny dark dots against the generally transparent body. Each one contains a handful of photoreceptor cells and a small amount of pigment that acts as a light shield, allowing the cell to detect the direction of incoming light. It is a beautifully simple sensory system, just enough to navigate a world where the most important information is whether it is light or dark outside. Some species that live in darkness have no eyes at all. While many tardigrade species possess simple eye spots, some have dispensed with eyes altogether. Species that live in permanently dark environments, such as deep soil layers, cave sediments, or the lightless depths of the ocean floor, have no need for light-sensing organs. Over evolutionary time, these populations have lost their eye spots entirely, a common pattern seen across many animal groups that colonize dark habitats. Without selective pressure to maintain them, the genes involved in eye spot development can accumulate mutations and eventually lose function. The result is a tardigrade that navigates its world entirely through touch and chemical sensing, relying on sensory structures in its cuticle to detect vibrations, moisture gradients, and the presence of food. For these eyeless species, the concept of light is irrelevant to survival. Their world is one of texture, chemistry, and contact, a landscape perceived through means that have nothing to do with vision. The loss of eyes is not a deficiency, but an adaptation, a streamlining of the body in response to an environment where eyes would serve no purpose. It is a quiet reminder that evolution shapes organisms to fit their world, not to match our expectations of what an animal should be. Tardigrades have no blood flowing through vessels. Unlike larger animals that pump blood through a network of arteries, veins, and capillaries, tardigrades have no circulatory system at all. There are no blood vessels, no red cells carrying oxygen, and no elaborate plumbing threading through their tiny bodies. This might seem like a significant limitation, but for an animal as small as a tardigrade, a dedicated circulatory system is simply unnecessary. Their bodies are so tiny that nutrients and gases can reach every cell without the need for a transport network. Instead, the fluids bathing their internal organs serve the function that blood performs in larger animals, delivering nutrients and oxygen directly to the tissues that need them. This open body plan is common among very small invertebrates and is a reflection of the physical principle that diffusion works efficiently over short distances. A tardigrade's entire body is essentially within diffusion range of its outer surface, meaning that oxygen absorbed through the skin can reach even the deepest internal cells within moments. It is a simple and elegant solution to the problem of internal transport, one that works perfectly at the scale of a creature smaller than a grain of sand. Their body cavity is filled with circulating fluid instead. In place of a true circulatory system, the interior of a tardigrade's body is filled with a fluid called hemolymph that bathes the organs directly. This fluid occupies the body cavity, known as a hemicole, and moves around as the animal walks, flexes its muscles, and changes position. The motion of the body itself helps to circulate the fluid, distributing nutrients from the gut to the muscles and other tissues. Hemolymph serves many of the same functions as blood in larger animals, carrying dissolved gases, nutrients, waste products, and immune cells throughout the body. It also plays a role in maintaining the body's internal pressure, which helps the tardigrade keep its shape and move its legs effectively. Because tardigrades are so small, this passive circulation is perfectly adequate for their needs. There is no pump driving the flow, no heart beating in the background, just the gentle sloshing of fluid as the animal goes about its daily business. It is a beautifully minimalist system, stripped of all unnecessary complexity and perfectly matched to the demands of life at the microscopic scale. They have no lungs or heart. The absence of a circulatory system in tardigrades extends to the organs that typically drive it. They have no heart to pump fluid and no lungs to exchange gases. In larger animals, the heart is essential for pushing oxygenated blood to distant tissues, and the lungs provide a large surface area for absorbing oxygen from the air. Tardigrades need neither. Their microscopic size means that every cell in their body is close enough to the outside environment to receive oxygen directly through the skin. The thin, permeable cuticle that covers their body allows oxygen to diffuse inward and carbon dioxide to diffuse outward without any specialized respiratory organ. This is only possible because of Fick's law of diffusion, which tells us that gas exchange is efficient over very short distances, but becomes inadequate as organisms grow larger. Tardigrades sit well within the range where simple diffusion is more than sufficient. The absence of a heart and lungs is not a sign of primitiveness, but rather an example of efficient biological design. When you are small enough, complexity becomes unnecessary and simplicity becomes the most elegant solution. Oxygen diffuses directly through their soft cuticle. The entire surface of a tardigrade's body acts as its respiratory system. The soft, flexible cuticle that forms their outer covering is thin enough to allow oxygen molecules to pass through it and into the underlying cells, while carbon dioxide produced by metabolism diffuses outward and is released into the surrounding water. This process requires no muscular effort and no specialized tissue. It happens passively, driven by the natural tendency of gas molecules to move from areas of high concentration to areas of low concentration. For this system to work, the tardigrade must remain in contact with water or a very thin film of moisture, because the cuticle needs to be damp for gas exchange to occur. This is why tardigrades are classified as aquatic animals even when they live on land, because they depend on the microscopic layer of water that clings to mosses, soil particles, and lichen surfaces. When this water evaporates and the environment dries out, gas exchange ceases, which is one of the triggers for the tardigrade to enter its dormant ton state. The simplicity of this respiratory method is a quiet marvel of miniature biology. Their brain layout echoes patterns seen in arthropods. Despite their tiny size, tardigrades possess a well-defined brain that shares structural similarities with the brains of arthropods such as insects and crustaceans. The tardigrade brain sits in the head region and is connected to a ventral nerve cord that runs along the underside of the body, sending branches to each pair of legs. This arrangement closely mirrors the nervous system layout found in many arthropod groups, providing further evidence for the evolutionary relationship between tardigrades and their larger cousins. The brain itself consists of clusters of nerve cells called ganglia, which process sensory information from the eye spots, the mouthparts, and the body surface. Additional ganglia are located along the nerve cord, one for each body segment, coordinating movement and local reflexes. For an animal measured in fractions of a millimeter, the complexity of this nervous system is impressive. It allows tardigrades to sense their environment, respond to stimuli, coordinate the movement of 8 legs, and make decisions about feeding, mating, and when to enter dormancy. The echoes of arthropod architecture in such a small body speak to the deep evolutionary connections that bind tardigrades to the wider world of jointed-legged animals. Females lay eggs with elaborately ornamented shells. Tardigrade eggs are among the most beautifully ornate structures in the microscopic world. Females produce eggs encased in shells that can be surprisingly elaborate, decorated with spikes, ridges, filaments, and intricate surface patterns that vary dramatically between species. These ornamentations are not merely decorative. They serve practical purposes, helping the eggs anchor to moss fibers, resist physical damage, and withstand the same environmental extremes that the adults can tolerate. Under a scanning electron microscope, tardigrade eggshells reveal a level of architectural detail that seems almost impossible for such a tiny organism to produce. Some species create eggs with long, branching spines that interlock with the surrounding substrate, while others produce smooth, round eggs with subtle surface textures. The shell itself is composed of tough chitin-like material secreted by the mother's reproductive system, providing a protective barrier around the developing embryo. In many species, the eggs are laid during molting, deposited inside the shed cuticle where they are sheltered until the young hatch. Each egg is a tiny masterpiece of biological engineering, perfectly adapted to give the next generation the best chance of survival. Some eggs sprout spikes that make them look like alien spheres. Among the most visually dramatic tardigrade eggs are those that bristle with long, sharp spikes, giving them an appearance that would not look out of place in a science fiction film. Under high magnification, these spiked eggs resemble tiny alien planets or the seed heads of some otherworldly flower, each spine precisely formed and evenly spaced around the surface. The spikes serve several functions. They help anchor the egg in place among moss fibers and soil particles, preventing it from being washed away by rain or dislodged by wind. They also increase the surface area of the egg, which may assist with gas exchange during embryonic development. In some species, the spikes are hollow, adding structural strength while minimizing weight. The variation in spike shape and density between different tardigrade species is so distinctive that researchers can often identify the species from the egg alone without ever seeing the adult. These extraordinary structures are produced by the female's reproductive tract as the eggshell is secreted, each spike formed with remarkable precision at the cellular level. It is one of the many small wonders that emerge when you look closely enough at the microscopic world. Mothers sometimes shed their skin to guard the eggs inside. In a number of tardigrade species, the female has evolved an unusual method of protecting her eggs. Rather than laying them freely into the environment, she deposits them inside her old cuticle as she molts. During the molting process, the tardigrade sheds its entire outer skin in one piece, and the eggs are released into this shed casing before it separates from the body. The discarded cuticle then acts as a protective capsule, shielding the eggs from desiccation, physical damage, and predators until they are ready to hatch. Inside this natural incubator, the embryos develop in relative safety, surrounded by the tough material of the mother's former skin. This strategy is particularly effective in harsh environments where exposed eggs might be vulnerable to drying or freezing. In species that practice this form of egg guarding, the shed cuticle can sometimes be found with multiple eggs nestled inside, each one developing at its own pace. It is a quietly ingenious form of parental care, one that requires no active effort from the mother and yet provides her offspring with a significant survival advantage. The molted skin becomes a final gift, a shelter built from her own body. Hatchlings emerge as tiny, fully formed tardigrades. When tardigrade eggs hatch, what emerges is not a larva or a grub, but a miniature, fully formed tardigrade, complete with all 8 legs, a functioning digestive system, and the same basic body plan as the adult. There is no metamorphosis, no dramatic transformation from one body form to another. The hatchling simply begins its life as a smaller version of its parent, ready to walk, feed, and explore its immediate surroundings from the very first moments of its existence. This mode of development, known as direct development, is common among tardigrades and contrasts with the larval stages seen in many other invertebrate groups. The young tardigrade will grow by molting, shedding its cuticle periodically and expanding slightly with each molt until it reaches adult size. Each molt is a vulnerable moment, as the new cuticle is briefly soft and the animal is exposed. But within hours, the new covering hardens and the tardigrade resumes its quiet routine. The fact that hatchlings are immediately functional means that every tardigrade, from the moment it breaks free of its egg, is already equipped with the survival tools that define its species. Some species show sex ratios strongly skewed toward females. In many tardigrade species, males are rare or even absent from natural populations, resulting in sex ratios that are overwhelmingly dominated by females. Some species reproduce through parthenogenesis, a process in which unfertilized eggs develop into viable offspring without any contribution from a male. In these populations, every individual is female, and males have never been observed despite extensive sampling. Other species do produce males, but in such low numbers that they are difficult to find among the far more abundant females. The reasons for this skewed ratio are not fully understood, but parthenogenesis offers clear advantages in unstable or isolated environments. A single female tardigrade, carried by the wind to a new moss patch, can establish an entire population without needing to find a mate. This reproductive flexibility has likely contributed to the success of tardigrades in colonizing such a wide range of habitats across the globe. In species where males do occur, mating has been observed to involve the male depositing sperm near the female or directly onto her body as she molts. But in many tardigrade communities, the quiet business of reproduction proceeds without males playing any role at all. They live on mountaintops in thin films of water. High on the world's tallest mountains, where the air is thin, temperatures plunge far below freezing, and ultraviolet radiation is intense, tardigrades have found a way to persist. They live in the thin films of water that cling to mosses and lichens growing on exposed rock surfaces, surviving at altitudes where most other animals cannot. Mountain tardigrades endure daily temperature swings that would kill less resilient creatures, freezing solid at night and thawing again as the sun returns. The winds at high altitude can desiccate their habitats within hours, but tardigrades simply enter their tun state and wait for the next rain or snowmelt. Some species found at extreme elevations have been shown to possess particularly robust DNA repair mechanisms, possibly as an adaptation to the increased ultraviolet exposure they face. Their presence on mountaintops is a testament to the versatility of the tardigrade body plan and the effectiveness of their dormancy strategy. Wherever there is a patch of moss and a film of water, tardigrades can establish a foothold, even on the roof of the world. They also thrive near deep-sea hydrothermal vents. At the opposite extreme from frozen mountaintops, tardigrades have also been found in the deep ocean, including in sediments near hydrothermal vents, where superheated water erupts from the seafloor. These vents create some of the most extreme environments on Earth, with temperatures that can exceed 300 degrees Celsius at the point of emission and intense chemical gradients in the surrounding water. Tardigrades living in the cooler zones around these vents inhabit a world of perpetual darkness, crushing pressure, and unusual chemistry. They feed on the bacterial mats and organic material that thrive in the nutrient-rich waters near the vents, carving out a niche in one of the most hostile habitats on the planet. Marine tardigrade species are generally different from their freshwater and terrestrial relatives, having adapted over millions of years to the specific challenges of ocean life. Their presence near hydrothermal vents is particularly noteworthy because it demonstrates that tardigrades can tolerate not just extreme temperatures and pressures, but also the unusual chemical conditions that surround these geological features. It is a quiet reminder that the tardigrades' range extends from the highest peaks to the deepest ocean floors. Populations persist inside polar ice and snow. In the frozen landscapes of the Arctic and Antarctic, where temperatures routinely plunge far below zero and daylight disappears for months at a time, tardigrades maintain viable populations within ice and snow. They have been found living in the thin layers of liquid water that exist within glacial ice, in meltwater pools on the surface of ice sheets, and in the moss and lichen that cling to exposed rock in polar regions. During the long polar winters, these tardigrades enter dormancy, freezing along with their environment and waiting in suspended animation for the brief summer thaw. When warmer temperatures return, meltwater seeps through the ice and rehydrates the dormant tons, and the tardigrades resume feeding and reproducing during the short window of activity available to them. Some polar tardigrade species have been found in ice cores drilled from glaciers, preserved at depths that suggest they have been frozen for considerable periods. Their ability to survive in these extreme cold environments makes them one of the few multicellular animals that can truly call the polar ice home. It is a life lived in the margins of possibility, measured in brief summers and long frozen winters. Tropical rainforests are particularly rich with tardigrade species. While tardigrades are famous for surviving in extreme environments, some of their greatest diversity is found in the warm, humid canopies of tropical rainforests. The mosses, lichens, and epiphytic plants that drape the branches of rainforest trees provide an ideal habitat for tardigrades, offering a constant supply of moisture, abundant food in the form of algal cells and bacteria, and a complex three-dimensional landscape to explore. Tropical forests support a remarkable variety of tardigrade species, many of which have yet to be formally described by science. The warmth and humidity of these environments mean that tardigrades can remain active for longer periods than in more seasonal habitats, allowing them to feed, grow, and reproduce at a faster pace. Canopy surveys using moss samples collected from high branches have revealed thriving communities of tardigrades living entirely above the forest floor, inhabiting a world suspended between earth and sky. The richness of tardigrade life in tropical forests is a reminder that these creatures are not just extremophiles, but also thrive in gentler environments where conditions are consistently favorable. Wherever moss grows and water gathers, tardigrades are likely to follow. They creep between individual grains of beach sand. The spaces between grains of sand on a beach, known to scientists as interstitial habitats, are home to a surprising variety of microscopic life, and tardigrades are among the most common inhabitants. In this miniature world, each gap between grains is a chamber, each channel a corridor, and each film of water a pathway along which tiny organisms can travel. Tardigrades move through these interstitial spaces using their clawed legs, gripping the surfaces of sand grains and pulling themselves along with their characteristic slow gait. The marine species found in beach sand are specially adapted to this environment, with body shapes and claw structures that help them navigate the narrow passages between particles. Some feed on the diatoms and bacteria that coat the surfaces of sand grains, while others are predators that hunt even smaller organisms in the same habitat. The interstitial world of beach sand is invisible to anyone walking across a shore, yet beneath every footstep, countless tardigrades may be going about their quiet lives. It is one of the most overlooked ecosystems on Earth, a hidden metropolis measured in micrometers and populated by creatures of astonishing resilience. Freshwater ponds and garden bird baths host hidden colonies. You do not need to travel to extreme environments to find tardigrades. They are living in quiet abundance in freshwater ponds, puddles, and even the bird baths in suburban gardens. Any body of still or slow-moving water that contains algae, moss, or organic sediment is likely to harbor a population of these microscopic wanderers. In a garden bird bath, the thin layer of green algae that forms on the inner surface is a potential feeding ground for tardigrades, providing both food and the moisture they need to remain active. Freshwater ponds, with their rich communities of microorganisms, offer an even more diverse habitat. Tardigrades in these environments feed on algal cells, bacteria, and sometimes on other tiny invertebrates. They may also enter dormancy if the pond dries up during a warm spell, waiting in their tun state until rain refills the water. The presence of tardigrades in such ordinary settings is a gentle reminder that the extraordinary is often hidden in the everyday. The next time you pass a garden pond or a neglected bird bath, you might pause to consider the invisible community of water bears quietly living within it. Moss and lichen carpets are their classic homes. Of all the habitats tardigrades occupy, mosses and lichens are the most iconic and the most commonly associated with these tiny creatures. The dense, spongy structure of a moss cushion creates a miniature forest of stems and leaves that trap moisture, providing the thin film of water tardigrades need to remain active. Lichens, those symbiotic partnerships between fungi and algae, offer a similar combination of moisture retention and abundant food. For a tardigrade, a patch of moss on a stone wall or a crust of lichen on a tree trunk is an entire world, rich with algal cells to feed on, sheltered from direct sunlight, and reliably damp after every rain. Scientists studying tardigrades often begin by collecting moss samples, soaking them in water, and examining the resulting suspension under a microscope. This simple technique reveals a surprising abundance of tardigrades in even the most unremarkable-looking patches of moss. The association between tardigrades and moss is so strong that the nickname moss piglet is a direct reflection of this relationship. It is a partnership that has endured for millions of years, the silent inhabitants and their green, spongy homes. Urban roofs and parking lots can harbor them. Tardigrades are not limited to wild or natural habitats. They have been found thriving in some of the most urbanized environments imaginable, including the mosses and lichens growing on city rooftops, the edges of parking lots, and the cracks in concrete pavements. Anywhere that a thin layer of organic material accumulates and retains moisture, tardigrades can potentially establish a population. Urban roofs, in particular, often develop patches of moss and lichen over time, and surveys of these seemingly barren surfaces have revealed diverse communities of tardigrades quietly living above the bustle of city life. The dust, pollution, and temperature extremes of urban environments do not deter them, thanks to their ability to enter dormancy whenever conditions become unfavorable. Some studies have even compared tardigrade diversity across different types of urban surfaces, finding that green roofs with deliberate plantings of moss and sedums support especially abundant populations. The presence of tardigrades in cities is a powerful illustration of how life finds its way into every available niche. Even in the most human-dominated landscapes, microscopic water bears are quietly going about their ancient business. They live on every continent, including Antarctica. Tardigrades are among the most widely distributed animals on Earth, found on every continent from the tropical forests of South America to the frozen wastes of Antarctica. Their global range is a direct consequence of their extraordinary survival abilities. Dormant tons, light enough to be carried by the wind, can travel vast distances through the atmosphere, landing on new moss patches, lichens, and soil surfaces far from where they originated. This wind-borne dispersal, combined with transport by rainwater, birds, and other animals, has allowed tardigrades to colonize virtually every terrestrial habitat on the planet. In Antarctica, they survive in the mosses and lichens that grow during the brief summer months, entering dormancy for the rest of the year. In tropical regions, they thrive year-round in the canopy and forest floor. In temperate zones, they cycle between activity and dormancy with the seasons. No major landmass is without its tardigrade population, and new species continue to be discovered in even the most well-studied regions. Their cosmopolitan distribution is a testament to the power of being small, tough, and patient enough to let the wind carry you wherever it will. Some species have been found in household gutters. The humble household gutter, often clogged with leaves, dirt, and decaying organic matter, turns out to be a surprisingly hospitable environment for tardigrades. The damp, nutrient-rich sludge that accumulates in gutters provides many of the same conditions found in natural moss and lichen habitats: moisture, organic food sources, and a sheltered microenvironment protected from direct sunlight and wind. Researchers sampling gutters on residential and commercial buildings have found thriving populations of tardigrades living among the debris, feeding on the algae and bacteria that colonize the accumulated sediment. During dry periods, these gutter-dwelling tardigrades simply enter dormancy, waiting for the next rainfall to reactivate their watery world. It is a testament to their adaptability that an animal whose lineage dates back over 500 million years can flourish in a structure built by humans just a few decades ago. The gutter tardigrade is a perfect symbol of how these creatures fit into any available niche, no matter how modest. If you have a house with gutters, there is a reasonable chance that tiny water bears are living in them right now, quietly enduring above your head without you ever knowing. They were the first animals to survive direct space exposure. In 2007, tardigrades made scientific history by becoming the first animals to survive direct, unprotected exposure to the vacuum and radiation of outer space. As part of a European Space Agency experiment, dehydrated tardigrades were placed on the exterior of a spacecraft orbiting the Earth and exposed to the full harshness of the space environment for 10 days. They endured the vacuum, cosmic radiation, and extreme temperature fluctuations without any protective shielding. When the spacecraft returned and the tardigrades were rehydrated back on Earth, a significant proportion revived and resumed normal biological functions. Some even went on to reproduce successfully. This result stunned the scientific community because no multicellular animal had ever been shown to tolerate such conditions before. The experiment confirmed that the tardigrade's ton state provides protection not just against terrestrial extremes, but against the conditions of outer space itself. It was a landmark moment in astrobiology, demonstrating that the boundary between the living world and the lifeless void of space is not as absolute as it might seem. The tiny water bear had quietly rewritten what we thought was possible for life. In 2007, orbital survivors revived after returning to Earth. The 2007 space experiment did not merely show that tardigrades could survive in orbit. It showed that they could come back to life afterwards. When the dried tardigrades were returned from the exterior of the Photon M3 spacecraft and gently rehydrated in a laboratory, many of them awoke from their dormancy and began moving, feeding, and behaving normally within hours. This revival was carefully documented, with researchers monitoring the animals for signs of damage, reduced fertility, or abnormal behavior. While some of the space-exposed tardigrades showed lower survival rates compared to control groups that had remained on Earth, the fact that any survived at all was extraordinary. Those that did revive appeared healthy and functional, their cells intact, and their biology seemingly unharmed by the ordeal. Some of these orbital survivors went on to lay viable eggs, producing offspring that showed no obvious ill effects from their parents' space exposure. The experiment demonstrated that the tardigrade's protective chemistry is robust enough to shield its cells not just from any single extreme, but from the combined assault of vacuum, radiation, and temperature extremes encountered simultaneously in low Earth orbit. Some returned from space and still reproduced successfully. Perhaps the most remarkable aspect of the tardigrade space experiments was that some of the survivors did not merely revive, but went on to reproduce. After being exposed to the vacuum and radiation of space for 10 days and then rehydrated on Earth, certain individuals laid eggs that hatched into healthy offspring. This means that the genetic material of these tardigrades, their DNA, was sufficiently intact after space exposure to support the production of a new generation. For any organism, the ability to reproduce after enduring extreme conditions is the ultimate test of survival. It is not enough to simply wake up. The cells must be functional at every level, from basic metabolism to the complex process of egg production and embryonic development. The fact that space-exposed tardigrades passed this test suggests that their protective proteins, particularly DSUP, and the intrinsically disordered proteins that form the vitrified matrix, do an extraordinary job of shielding the most critical cellular structures. These results have profound implications for astrobiology, suggesting that microscopic life could potentially survive journeys through space and remain reproductively viable upon arrival at a new world. They endured cosmic radiation on low Earth orbit platforms. In low Earth orbit, beyond the protective shield of our atmosphere, cosmic radiation is a constant and dangerous presence. Charged particles from the Sun and from distant supernovae stream through space at enormous speeds, and without the filtering effect of the atmosphere, they strike anything in their path with full force. Tardigrades placed on external platforms of the International Space Station and other orbital facilities have been directly exposed to this bombardment. The radiation they encountered included ultraviolet rays, X-rays, and high-energy charged particles, all of which are capable of shredding DNA and destroying cellular structures. Yet many tardigrades survived the exposure, particularly those shielded from the harshest ultraviolet wavelengths by thin filters. Their survival is attributed to the same combination of DSUP protein protection, DNA repair enzymes, and the compact TUN state that protects them against radiation on Earth. These orbital experiments have helped scientists understand the upper limits of tardigrade radiation tolerance and have provided valuable data for researchers studying how life might endure beyond our planet. Each surviving tardigrade represented a small victory for biology against the seemingly hostile emptiness of space. Thousands were accidentally delivered to the moon in a crashed spacecraft. In 2019, an Israeli spacecraft called Bereshit attempted to land on the moon, but crashed on the lunar surface during its descent. On board were thousands of dehydrated tardigrades, placed there as part of the Arch Mission Foundation's project to create a backup of Earth's biology in space. When the craft hit the moon at high speed, its contents were scattered across the impact site. The tardigrades, sealed in resin and in their dried TUN state, were likely dispersed with the wreckage. Whether any of them could have survived the crash impact is uncertain. Studies on impact tolerance suggest that at the velocities involved, The forces may have exceeded what even tardigrades can endure. But the incident captured the public imagination and raised interesting questions about planetary contamination and the unintentional spread of Earth life to other worlds. If any tardigrades did survive intact, they would remain in their dormant state on the airless, waterless lunar surface, unable to revive without liquid water. They would be tiny, frozen time capsules, waiting in silence on a world where rain will never fall. They are nicknamed astronauts of the microscopic world. Given their proven ability to survive in space, their accidental delivery to the moon, and their inclusion in numerous orbital experiments, tardigrades have earned the affectionate nickname of astronauts of the microscopic world. It is a title that captures both their size and their extraordinary accomplishments. No other microscopic animal has been so thoroughly tested against the rigors of space, and none has performed so impressively. The nickname has taken hold in popular science writing and across media coverage, helping to transform tardigrades from obscure laboratory subjects into household names. There is something deeply appealing about the image of a tiny, chubby water bear floating through the cosmos, enduring conditions that would destroy almost any other living creature. The nickname also reflects a broader truth about tardigrades, that their survival abilities place them in a category of their own, separate from all other animals. They are not merely tough terrestrial creatures, but organisms whose biology has been tested against the most extreme environment known, the vacuum of space, and found to be sufficient. In the quiet halls of science, the microscopic astronaut walks on. Models suggest they could survive certain asteroid impacts. Theoretical models examining the resilience of tardigrades have suggested that they could survive certain types of asteroid impacts, provided the conditions fall within specific limits. Researchers have calculated that while a catastrophic impact large enough to boil the oceans would destroy tardigrades along with everything else, smaller impacts that cause severe but localized damage might leave deep ocean tardigrade populations intact. The key factor is whether the impact generates enough heat and pressure to sterilize every habitat on the planet simultaneously. For all but the very largest asteroid strikes, pockets of survivable conditions would likely persist in the deep sea, underground, or at the poles, where tardigrades could endure in their ton state until conditions improved. These calculations were part of a broader study into which organisms might survive global catastrophes, and tardigrades consistently emerged as among the most likely survivors. The models suggest that it would take an extraordinarily rare event, far more severe than any impact recorded in the last several 100 million years, to eliminate tardigrades entirely. It is a comforting thought that somewhere on our planet, these tiny creatures would likely persist through all but the most absolute of calamities. Their ton state informs ideas about interstellar survival. The remarkable ability of tardigrades to enter a state of near-complete metabolic shutdown has inspired scientists to think more seriously about how life might survive the vast distances between stars. Interstellar space is unimaginably hostile, filled with cosmic radiation, near absolute zero temperatures, and a vacuum more perfect than anything achievable on Earth. Yet the tardigrade ton state offers a proof of concept that biological systems can endure such conditions, at least for limited periods. If microscopic organisms could remain dormant for thousands or millions of years while embedded in rock fragments ejected from planets by asteroid impacts, they might theoretically survive a journey from one star system to another. This idea, known as panspermia, has been debated for over a century, but tardigrade research has given it new scientific credibility. The TUN's combination of desiccation tolerance, radiation resistance, and metabolic shutdown makes it one of the closest models we have for what interstellar biological survival might look like. While no tardigrade has yet been tested over the timescales required for interstellar travel, their demonstrated tolerance sets a benchmark for what biology is capable of. They feature in panspermia experiments testing life's limits. Panspermia is the hypothesis that life can be transported between planets or even between star systems aboard fragments of rock, dust, or ice. Tardigrades have become central to experimental tests of this idea because their extreme tolerance makes them ideal candidates for simulating the conditions such a journey would involve. Researchers have subjected tardigrades to simulated meteorite impacts, prolonged vacuum exposure, intense radiation, and extreme temperatures, all in an effort to determine whether any multicellular organism could survive the stages of planetary ejection, transit through space, and arrival on a new world. The results have been mixed but encouraging. Tardigrades can survive individual stresses that mimic parts of the journey, though the cumulative effect of all stresses combined over millions of years remains untested. These experiments have contributed valuable data to the broader debate about whether life on Earth could have originated elsewhere, or whether Earth life might already have been inadvertently scattered to other bodies in our solar system. Tardigrades may never have made such a journey naturally, but they have shown us that the idea is not as far-fetched as it once seemed. They help scientists design experiments for Mars and beyond. The search for life beyond Earth is one of the most ambitious goals of modern science, and tardigrades are playing a quiet but important role in shaping that search. By studying what tardigrades can and cannot survive, scientists gain insights into the kinds of conditions that might support life on other worlds. If a microscopic animal on Earth can tolerate radiation levels comparable to those on the Martian surface, or pressures similar to those on icy moons, then the possibility that similar organisms might exist in those environments becomes a little more plausible. Tardigrade research also helps engineers design life detection instruments for space missions by identifying the biological signatures that extremophile organisms leave behind. Understanding the limits of tardigrade survival helps define the boundaries within which we should search for extraterrestrial biology. Some researchers have even proposed sending tardigrades to Mars deliberately, as a way of testing whether Earth organisms can survive on the Martian surface under real conditions. Whether or not this ever happens, the humble tardigrade has become an invaluable reference point for astrobiology, a living yardstick against which we measure the potential for life elsewhere in the cosmos. Tardigrades can repair severely damaged DNA. One of the most remarkable abilities of tardigrades is their capacity to repair DNA that has been severely damaged by radiation, desiccation, or other environmental stresses. In most organisms, extensive DNA damage triggers cell death because the molecular machinery cannot keep up with the scale of the destruction. Tardigrades, however, possess unusually efficient DNA repair pathways that can stitch broken strands back together and correct errors that would overwhelm the repair systems of other animals. When a tardigrade revives from its TUN state, its cells must contend with accumulated DNA damage from the period of dormancy, and they do so with impressive speed and accuracy. Studies have shown that tardigrades can tolerate doses of ionizing radiation that shatter their DNA into fragments, and yet within hours of being returned to normal conditions, much of this damage is repaired. The mechanisms involved are still being studied, but they include both well-known repair enzymes found in other organisms and unique tardigrade-specific proteins that enhance the process. This extraordinary repair capacity is one of the key pillars of tardigrade resilience, ensuring that even after prolonged exposure to destructive forces, the genetic blueprint of the animal remains functional. A unique protein called DSUP shields their DNA from harm. Among the many molecular tools in the tardigrade survival kit, one stands out for its sheer novelty. A protein called DSUP, short for damage suppressor, is found exclusively in tardigrades and has no known equivalent in any other organism. DSUP works by binding directly to DNA and forming a protective cloud around the genetic material, physically blocking radiation and reactive molecules from reaching the vulnerable strands. When researchers introduced the gene for DSUP into cultured human cells, those cells showed a remarkable increase in resistance to X-ray radiation, suffering roughly half the DNA damage of unprotected cells. This experiment demonstrated that DSUP's protective function is transferable, meaning it works even in a completely different biological context. The discovery of DSUP opened up exciting possibilities for biotechnology, from protecting crops against radiation to shielding astronaut cells during long-duration space missions. The protein's mechanism is elegant in its simplicity. Rather than repairing damage after the fact, it prevents the damage from occurring in the first place. DSUP is a product of hundreds of millions of years of tardigrade evolution, a molecular shield forged in the crucible of extreme survival. That protein has protected cultured human cells in laboratory experiments. The discovery that DSUP could protect human cells was one of the most exciting findings to emerge from tardigrade research. In a landmark experiment, scientists took the gene encoding DSUP from a tardigrade genome and inserted it into human cell cultures. The modified cells were then exposed to X-ray radiation alongside unmodified control cells. The results were striking. Cells carrying the DSUP gene sustained significantly less DNA damage than their unprotected counterparts, demonstrating that this tardigrade protein could function effectively within a completely different organism's cellular machinery. The experiment showed that DSUP's protective mechanism is not specific to tardigrade cells, but is broadly compatible with mammalian biology. This has profound implications for medicine and biotechnology. If DSUP or proteins derived from it could be used to protect human cells in vivo, it might one day help shield patients undergoing radiation therapy, protect workers in radiation-intensive industries, or even safeguard astronauts during long missions beyond the Earth's magnetosphere. While such applications remain in the future, the successful transfer of tardigrade protection to human cells was a powerful demonstration that nature's solutions, honed over millions of years, can sometimes be borrowed and applied in entirely new contexts. During dormancy, their cells enter a glass-like vitrified state. When a tardigrade enters its tun state, the interior of its cells undergoes a remarkable transformation. As water is expelled from the body, special proteins and sugars produced by the tardigrade cells fill the spaces that water once occupied. These molecules form a dense, glass-like matrix that encases the cell's internal structures, including membranes, enzymes, and DNA, in a rigid but non-crystalline solid. This process is called vitrification, and it is fundamentally different from freezing. When water freezes, it forms sharp ice crystals that can puncture and destroy cell membranes. Vitrification avoids this by creating an amorphous solid with no crystal structure at all. The result is a cell that is locked in place, its delicate components held in position by a glassy scaffold that prevents them from collapsing, aggregating, or being damaged by the absence of water. This vitrified state can persist for years or even decades, maintaining the cell in a condition from which it can be revived simply by adding water. The glass dissolves, the structures are released, and normal cellular function resumes. It is one of the cleverest survival strategies in all of biology, and it is unique to organisms like tardigrades that have mastered the art of anhydrobiosis. Their DNA is packed tightly to reduce radiation damage. When tardigrades enter dormancy, their DNA undergoes a process of compaction that reduces its vulnerability to radiation damage. In an active cell, DNA is relatively loosely arranged, spread out through the nucleus in a form that allows genes to be read and copied. But in the TUN state, the DNA is packed far more tightly, condensed into a compact configuration that presents a smaller target for incoming radiation. The logic is simple. The less surface area exposed, the fewer hits the DNA is likely to take from ionizing particles or ultraviolet photons. This compaction works in concert with the DSUP protein, which wraps around the condensed DNA and provides an additional layer of physical shielding. Together, these two mechanisms dramatically reduce the amount of damage that the tardigrade's genetic material sustains during periods of dormancy. When the tardigrade revives, the DNA relaxes back into its active configuration, and any residual damage is repaired by the cell's efficient repair enzymes. This multi-layered defense strategy, combining physical compaction, protein shielding, and post-revival repair, is one of the reasons tardigrades can tolerate radiation doses that would be lethal to almost any other animal. Their genomes contain genes found in no other known organism. Sequencing the genomes of tardigrade species has revealed something truly unusual. Alongside the familiar genes shared with other animals, tardigrade genomes contain a significant number of genes that have no counterpart in any other known organism. These orphan genes, as they are sometimes called, appear to be unique innovations of the tardigrade lineage, evolved over hundreds of millions of years to serve functions that no other animal requires. Many of these unique genes are involved in stress tolerance, encoding the intrinsically disordered proteins and other protective molecules that give tardigrades their extraordinary resilience. Others have functions that are not yet fully understood, hinting at aspects of tardigrade biology that remain to be explored. The presence of so many unique genes is unusual for any animal and reflects the extreme evolutionary path that tardigrades have followed. While most animal lineages share the vast majority of their genes through common ancestry, tardigrades have developed an additional toolkit of molecular innovations found nowhere else in the tree of life. These unique genes are a genetic signature of the tardigrade's singular approach to survival, a biological fingerprint of resilience. They switch off most metabolic processes while in a TUN. When a tardigrade enters the TUN state, it does not merely slow down its metabolism. It switches off almost every metabolic process entirely. The chemical reactions that drive growth, reproduction, movement, digestion, and even basic maintenance of cellular structures all cease, or drop to levels so low they are virtually undetectable by current scientific instruments. Energy consumption falls to near zero because there is no water available to serve as a medium for biochemical reactions and no active molecular machinery to consume it. In this state, the tardigrade is not truly alive in the conventional sense, yet it is not dead either. It exists in a gray zone between the two, a state of biological suspension that can last for years or decades. The moment water returns, the metabolic machinery restarts with remarkable efficiency. Enzymes become active, membranes regain their fluidity, and the complex web of chemical reactions that sustains life begins to hum once more. This ability to toggle metabolism on and off like a switch is one of the most extraordinary features of tardigrade biology and one that continues to fascinate researchers studying the fundamental nature of life. Some species stockpile trehalose to stabilize cell structures. Trehalose is a sugar molecule that plays a crucial role in the survival strategy of many desiccation-tolerant organisms, and certain tardigrade species produce it in abundance when preparing to enter dormancy. As the tardigrade dehydrates, trehalose molecules replace the water that normally surrounds proteins, membranes, and other cellular structures. The sugar forms hydrogen bonds with these molecules, holding them in their correct shapes even after all the water has gone. Without trehalose, the delicate folded structures of proteins would collapse and cell membranes would fuse and lose their integrity. With it, the cell's architecture is preserved in a kind of sugar glass, stable and protected until rehydration occurs. Not all tardigrade species rely on trehalose to the same degree. Some produce very little of it and instead depend more heavily on their unique intrinsically disordered proteins for protection. But in those species that do use trehalose, the sugar is an essential component of the survival toolkit, produced on demand as conditions deteriorate. It is one of nature's simplest, yet most effective preservation agents, a sweet shield against the destruction of drying. Other species use sugars that act as natural antifreeze. In addition to trehalose, some tardigrade species produce other sugars and sugar alcohols that function as natural antifreeze agents, lowering the freezing point of their body fluids and preventing the formation of damaging ice crystals within their cells. When temperatures drop, these cryoprotectant molecules accumulate in the cytoplasm, disrupting the orderly arrangement of water molecules that is necessary for ice crystal formation. By keeping the internal environment in a liquid or glassy state, even at sub-zero temperatures, these sugars protect the delicate membranes and proteins that are essential for the cell's survival. The strategy is similar to the one used by many cold-adapted insects and fish, but tardigrades have refined it to work in concert with their broader dehydration tolerance. Some species combine antifreeze sugars with vitrification proteins, creating a double layer of protection that makes them resistant to both freezing and drying simultaneously. This versatility is part of what makes tardigrades so successful in cold environments, from alpine peaks to polar ice. Their chemistry is a quiet masterclass in preparation, a set of molecular tools assembled before the cold arrives and ready to perform the moment they are needed. Their chromosome numbers vary widely between species. Across the many species of tardigrades described by science, chromosome numbers show a remarkably wide range of variation. Some species have as few as 5 or 6 chromosomes, while others carry 12 or more. This variation is unusually large for a single phylum and suggests a complex evolutionary history involving events such as genome duplication, chromosome fusion, and fragmentation. In some cases, closely related species have very different chromosome numbers, indicating that chromosomal rearrangements have occurred relatively recently in evolutionary terms. This variability makes tardigrades an interesting subject for geneticists studying how chromosome structure evolves and how changes in chromosome number affect an organism's biology. Despite these differences, all tardigrade species share the same fundamental body plan and the same core survival strategies, suggesting that the essential genes are maintained regardless of how they are distributed across chromosomes. The study of tardigrade chromosomes is still in its early stages, as many species have not yet been karyotyped. As more data become available, the patterns of chromosomal diversity in tardigrades may reveal new insights into the evolutionary forces that have shaped this ancient and resilient group. In good conditions, they reproduce multiple times across a short life. When conditions are favorable, with plenty of moisture, abundant food, and moderate temperatures, tardigrades can reproduce multiple times during their relatively brief active lives. Most tardigrade species have lifespans of only a few months to a couple of years when continuously active, and in that time, females may lay several clutches of eggs. Each clutch can contain anywhere from a handful to several dozen eggs, depending on the species. The speed of reproduction allows tardigrade populations to grow rapidly when their environment is supportive, quickly filling available habitat with new individuals. This reproductive strategy makes sense for animals living in unpredictable environments where conditions can change suddenly. By reproducing frequently during good times, tardigrades maximize the number of offspring that have a chance to establish themselves before the next drought, freeze, or other disturbance forces the population back into dormancy. In species that reproduce through parthenogenesis, this rapid multiplication is even more efficient, since every individual is a female capable of producing offspring. The combination of fast reproduction in favorable conditions and extreme tolerance when conditions worsen is a powerful survival formula that has served tardigrades well for hundreds of millions of years. Some species complete their entire life cycle in just weeks. In particularly active species living under optimal conditions, the entire life cycle of a tardigrade, from egg to adult to egg-laying parent, can be completed in a matter of weeks. This rapid turnover means that multiple generations can pass through a population in a single season, allowing for swift adaptation and population growth. The eggs hatch into miniature adults that begin feeding immediately, growing through a series of molts until they reach reproductive maturity. In warm, moist environments with plentiful food, each stage of development proceeds quickly and the interval between molts is short. This brisk pace of life stands in stark contrast to the seemingly endless patience of the tun state, where a single individual can persist for decades without any biological activity at all. The tardigrade life strategy is one of extremes: rapid development when conditions allow, and infinite patience when they do not. This flexibility gives tardigrades an enormous advantage in habitats that alternate between periods of plenty and periods of hardship. By compressing their life cycle into the shortest possible window, they ensure that their genes are passed on before the next environmental challenge arrives. Rehydrated tuns can reanimate within hours. When a dried tardigrade tun is exposed to water, the transformation from dormant capsule to active animal can occur with surprising speed. Within minutes, the tun begins to absorb water, swelling as moisture seeps through the cuticle and into the cells. The glassy matrix of protective proteins and sugars dissolves, releasing the membranes and organelles it had held in place. Over the next few hours, the tardigrade's body gradually returns to its original shape. The legs extend, the internal organs resume their normal proportions, and the first signs of movement appear. Metabolic activity restarts as enzymes become functional again in the watery environment they require. In many cases, a fully reanimated tardigrade is walking and feeding within just a few hours of rehydration. This rapid revival is a critical survival advantage because it allows tardigrades to take immediate advantage of transient moisture events such as brief rainfalls or morning dew. Every drop of water is an opportunity, and the faster a tardigrade can reactivate, the more time it has to feed and reproduce before conditions dry out again. The speed of this process never fails to impress those who witness it for the first time. Individuals can enter and exit dormancy repeatedly throughout life. Tardigrades do not have a single chance at dormancy. They can enter and exit the tun state multiple times throughout their lives, switching between active and dormant phases as conditions demand. Each time the environment dries out, freezes, or becomes otherwise hostile, the tardigrade can curl back into its ton and wait. When conditions improve, it revives and picks up where it left off. This ability to toggle between states is not without cost, as each cycle of desiccation and rehydration places some stress on the animal's cells and may contribute to gradual wear over time. Nonetheless, tardigrades are remarkably resilient to repeated cycling, and many individuals go through dozens of dormancy events during their lifetimes without obvious ill effects. This flexibility is essential for survival in the fluctuating microhabitats that tardigrades typically occupy, where conditions can change from wet to dry within hours. The capacity for repeated dormancy means that a tardigrade's effective lifespan can be extended far beyond what its active biology would normally allow. By pausing the clock whenever conditions worsen, tardigrades stretch their brief lives across spans of time that far exceed those of other animals their size. A few documented individuals have lived for over 3 decades. While most tardigrades have active lifespans measured in months, the inclusion of dormancy periods can extend their total existence dramatically. There are documented cases of individual tardigrades surviving for over 30 years, spending the vast majority of that time in a dried ton state and reviving when rehydrated decades later. In one well-known example, tardigrades recovered from a dried moss sample stored in a museum collection for more than 30 years, and some of these individuals revived and even produced viable offspring. The key to this longevity is the near-total shutdown of metabolism during dormancy. With no active biological processes running, there is very little wear and tear on the cells, and the aging process effectively pauses. The protective chemistry of the ton state preserves cellular structures with such fidelity that even after decades, the essential machinery of life remains functional. This extended survival raises interesting questions about how we define lifespan and aging. If an organism can pause its biology for years at a time, is it truly alive during those intervals? And does its age include the time spent in suspension? Tardigrades blur the boundaries of these concepts in the most quiet and unassuming way. They show measurable signs of aging over time. Despite their impressive longevity in dormancy, tardigrades are not immune to the passage of time. When they are active, feeding, and moving about their normal lives, they show measurable signs of aging just like other animals. Their cuticles may become less flexible, their movements slower, and their reproductive output may decline as they grow older. In laboratory studies, researchers have tracked individual tardigrades over their entire active lifespans and observed gradual declines in vigor and fecundity that are consistent with biological aging. The molecular mechanisms behind tardigrade aging are not yet fully understood, but they likely involve the same kinds of cumulative damage to DNA, proteins, and cellular structures that drive aging in other organisms. What makes tardigrades unusual is not that they avoid aging altogether, but that they can effectively pause it during dormancy. When active, they age. When dormant, the clock stops. This unique relationship with time gives tardigrades a form of conditional immortality, where the total duration of existence depends on how much time is spent in suspension versus how much is spent in active life. Even tardigrades ultimately die of old age. For all their extraordinary resilience, tardigrades are mortal creatures. They can survive extremes that would destroy almost any other animal, but they cannot cheat death forever. When continuously active without entering dormancy, most tardigrade species live for only a few months to a couple of years before dying of old age. Their bodies eventually wear out, their cells accumulate damage that even their impressive repair mechanisms cannot fully reverse, and they quietly cease to function. Even individuals that have spent decades in dormancy will eventually die of old age once they resume active life and exhaust their remaining biological lifespan. This mortality is an important reminder that tardigrade resilience is not the same as invincibility. Their survival strategies are about enduring hostile conditions long enough to resume normal life when things improve, not about living forever. Death is as much a part of the tardigrade story as survival. It is the quiet end that awaits every water bear after its slow walk across the moss is done, a natural conclusion to a life lived with extraordinary patience and remarkable endurance. Protozoans and nematodes commonly prey on them. Tardigrades may be famously tough, but in their active state, they are far from invincible against predators. Among their most common enemies are protozoans and nematode worms, both of which share the same microscopic habitats and are well equipped to hunt and consume tiny animals. Some protozoans, single-celled organisms that are among the most effective predators of the microscopic world, can engulf tardigrades whole or pierce their cuticles to extract nutrients. Nematode worms, which are abundant in virtually every soil and water habitat on Earth, include predatory species that actively hunt and devour tardigrades when they encounter them. These predator-prey interactions play an important role in regulating tardigrade populations in the wild, preventing any single species from dominating its habitat unchecked. The threat from predators may also be one of the selective pressures that drove the evolution of tardigrade dormancy. By entering the tun state, a tardigrade becomes a tough, desiccated pellet that is far less attractive and harder to consume than an active, water-filled animal. Dormancy, in this light, may serve not only as a defense against environmental extremes, but also as a way to become less conspicuous to hungry neighbors. Tiny beetle larvae are also known predators. Beyond protozoans and nematodes, tardigrades face predation from some rather unexpected sources, including the larvae of certain tiny beetles. These beetle larvae, themselves small enough to share the same moss and soil habitats as tardigrades, are active hunters that seek out microscopic prey. Their mouthparts are adapted for grasping and piercing small animals, and tardigrades are among the creatures on their menu. The interaction between beetle larvae and tardigrades takes place in a world measured in millimeters, a miniature ecosystem where even the predators are barely visible to the naked eye. For a tardigrade, encountering a beetle larva in the damp corridors of a moss cushion would be a serious threat, as the larva's larger size and more aggressive feeding behavior give it a decisive advantage. These predatory interactions are part of the rich and complex food web that exists at the microscopic scale, a web that includes herbivores, carnivores, parasites, and decomposers, all living within a few square centimeters of habitat. The tardigrade's role in this web is both predator and prey, a position that speaks to the ecological complexity of even the smallest environments. Certain fungi can infect and kill them. Tardigrades are not only vulnerable to animal predators, but also to microscopic parasites, including certain species of fungi that can infect and kill them. These parasitic fungi are adapted to target small invertebrates, invading their bodies through the cuticle and growing inside them, eventually consuming the host from within. Infected tardigrades may show visible signs of fungal growth, with hyphae, the thread-like structures of fungi, spreading through their body cavity and disrupting their internal organs. The infection is typically fatal, and the fungus may eventually produce spores on or near the dead tardigrade's body, ready to infect new hosts. This type of parasitism is well documented among other microscopic invertebrates, particularly nematodes, and it appears that tardigrades are not exempt from the same ecological pressures. The tun state may offer some protection against fungal infection, as the desiccated, metabolically inactive state is less hospitable to fungal growth, but active tardigrades, with their moist, nutrient-rich bodies, can fall victim to these silent pathogens. It is a reminder that in the microscopic world, survival is a constant negotiation with a wide array of threats, not just the physical extremes for which tardigrades are famous. Many species pierce algal cells with needle-like mouth parts. Tardigrades feed using a highly specialized structure known as a stylet, a sharp, needle-like mouthpart that they use to puncture the cells of algae, mosses, and other small organisms. The stylet is housed inside a tubular mouth called the buccal apparatus, and when the tardigrade feeds, it extends the stylet outward and drives it into the wall of a target cell like a microscopic syringe. Once the cell is pierced, the tardigrade uses a muscular pump in its pharynx to suck out the contents, extracting the nutrient-rich cytoplasm and leaving behind the empty cell wall. This feeding method is efficient and well-suited to the tardigrade's habitat, where algal cells and plant cells are the most readily available food sources. Under the microscope, the feeding process can be observed in real time, with the stylet visibly penetrating the cell and the gut gradually filling with the green contents of the prey. The precision with which tardigrades locate and pierce individual cells is remarkable for an animal with such a simple nervous system. It is a small but fascinating example of how even microscopic creatures can be highly effective predators within their own tiny world. They regrow their piercing stylets after each molt. The sharp stylets that tardigrades use to pierce cells are not permanent structures. Each time a tardigrade molts, shedding its old cuticle to allow for growth, the stylets are lost along with the rest of the outer covering. After molting, the tardigrade must grow an entirely new set of stylets before it can feed again. This process takes a short period, during which the animal is unable to puncture cells and must wait until the new mouthparts are fully formed and hardened. The replacement stylets are produced by specialized cells in the buccal apparatus, and they are assembled with the same precision as the originals, sharp enough to penetrate the walls of plant and algal cells. This cycle of loss and regrowth happens every time the tardigrade molts, which can occur several times during its active lifespan. For researchers, the shape and structure of the stylets are important taxonomic features used to identify different tardigrade species. The fact that these delicate instruments must be rebuilt from scratch with every molt speaks to the remarkable regenerative capacity of even the simplest multicellular animals. Plant-eating species graze on mosses and lichens. A large proportion of tardigrade species are herbivores, feeding primarily on the cells of mosses, liverworts, and lichens that form their primary habitat. These plant-eating tardigrades use their stylets to puncture individual cells and suck out the contents, grazing slowly across the surface of a moss leaf or lichen thallus as they go. Their feeding is selective, with individuals often targeting particular cell types or plant structures that offer the richest nutritional reward. Some species specialize in feeding on the photosynthetic cells of moss leaves, while others prefer the fungal or algal components of lichen. This herbivorous lifestyle places tardigrades in an important ecological role within their microhabitat, as they help regulate the growth of the plants and algae that make up the moss community. By consuming cells, they recycle nutrients and create space for new growth, contributing to the health and turnover of the moss ecosystem. Their grazing is gentle and slow, perfectly matched to the pace of their tiny world. It is a quiet form of gardening carried out by creatures too small to see, shaping the landscape one cell at a time. Others prefer to feed on bacterial biofilms. Not all tardigrades are plant eaters. Some species have adapted to feed primarily on bacterial biofilms, the thin, slimy layers of bacteria that coat surfaces in moist environments. These biofilms are found on rocks, soil particles, plant surfaces, and virtually any other substrate that remains damp for extended periods. For a tardigrade, a bacterial biofilm is a rich and reliable food source, providing a concentrated layer of nutritious cells that can be harvested by piercing individual bacteria or scraping across the surface with their mouthparts. Species that specialize in bacterial feeding often have slightly different stylet structures compared to their plant-eating relatives, adapted for penetrating the smaller, tougher cells of bacteria. Bacterial biofilms are ubiquitous in the environments where tardigrades live, ensuring a constant supply of food even when alga or plant cells are scarce. This dietary flexibility is part of what makes tardigrades so successful across such a wide range of habitats. Whether the available food is a moss leaf, a lichen crust, or a thin film of bacteria on a pebble, there is likely a tardigrade species adapted to exploit it. Several species are carnivorous hunters. While many tardigrades are peaceful herbivores, some species have taken a more aggressive path, evolving into active carnivores that hunt and consume other microscopic animals. These predatory tardigrades target a range of tiny prey, including rotifers, nematodes, and even other tardigrade species. They use their stylets not just to pierce plant cells, but to attack and immobilize small animals, puncturing their body walls and sucking out the internal fluids. Carnivorous tardigrade species tend to be among the larger members of the phylum, with more robust mouthparts and a more active hunting style than their herbivorous relatives. Under the microscope, a predatory tardigrade pursuing its prey is a dramatic sight, a slow-motion chase through the tangled corridors of moss and soil. The existence of carnivorous tardigrades adds another dimension to the ecological role of these tiny animals, placing them at multiple levels of the food web within their microhabitat. They are not simply passive grazers, but active participants in a complex community of predators and prey, all playing out on a scale invisible to the naked eye. Some will even prey on smaller tardigrades. Among the more striking examples of tardigrade carnivory is the fact that some species prey on other tardigrades. In the microscopic world, where food sources are scarce and competition is fierce, eating a smaller member of your own phylum can be a viable survival strategy. Larger predatory tardigrade species have been observed attacking and consuming smaller individuals, using their stylets to pierce the cuticle of their victim and drain the body contents. This form of intrigued predation is well known in many animal groups and reflects the intense selection pressures that operate even at the smallest scales of life. For the victim, there is little defense other than the ton state, which makes the body tougher and less appetizing. For the predator, a smaller tardigrade represents a high-quality meal, rich in proteins and lipids that are directly usable by a similar organism. These predatory encounters are a reminder that the peaceful image of the slow-stepping water bear does not tell the whole story. Within the moss and lichen where they live, tardigrades occupy a range of ecological niches, and not all of their interactions with their neighbors are gentle. A muscular pharyngeal bulb pumps food like a syringe. At the heart of the tardigrade's feeding apparatus is a powerful muscular structure called the pharyngeal bulb. This rounded, pump-like organ sits just behind the mouth and works on the same principle as a syringe. When the tardigrade pierces a cell with its stylet, the muscles of the pharyngeal bulb contract and then relax, creating a suction force that draws the cell contents up through the narrow stylet tube and into the gut. The pumping action is rhythmic and efficient, allowing the tardigrade to extract nutrients from a pierced cell in a matter of seconds. Under the microscope, the pharyngeal bulb can often be seen pulsing as the animal feeds, a tiny biological pump operating with quiet precision. The strength of this pump is impressive relative to the tardigrade's size, generating enough suction to overcome the resistance of viscous cell contents and pull them through an opening measured in micrometers. The pharyngeal bulb is one of the most distinctive anatomical features of tardigrades and is used alongside claw shape and stylet structure as a key characteristic for species identification. It is a small but vital piece of engineering that powers the tardigrade's entire nutritional life. You can watch their guts fill in real time under a microscope. One of the most delightful aspects of studying tardigrades under a microscope is the ability to watch their digestive process unfold in real time. Because many species have transparent bodies, the gut is clearly visible as a tube running through the center of the animal. When a tardigrade feeds on an algal cell, the bright green contents of the prey can be seen flowing through the stylet and into the pharyngeal bulb, then passing down into the gut, where they slowly change color as digestion proceeds. Watching a tardigrade eat is like peering through a window into a living machine, every stage of the process visible from the outside. Researchers use this transparency to study feeding rates, food preferences, and digestive efficiency without needing to dissect or harm the animal. For amateur microscopists and science enthusiasts, the sight of a tardigrade's gut filling with color is often the moment when these tiny creatures first come alive as real, dynamic animals rather than abstract scientific subjects. It turns a static image into a story, a narrative of hunger, pursuit, and satisfaction playing out on a scale measured in fractions of a millimeter. In a ton, they stop, feeding entirely for years. When a tardigrade enters its ton state, all feeding ceases completely. The stylets are retracted, the pharyngeal bulb becomes inactive, and the gut empties as the body dehydrates and contracts. For the entire duration of dormancy, which can last from weeks to decades, the tardigrade consumes nothing at all. There is no need for food because there is no metabolism to fuel. The energy reserves stored in the body before dormancy began are preserved in the vitrified state, waiting to be mobilized when the tardigrade rehydrates and resumes active life. This ability to go without food for such extended periods is part of what makes the ton state so effective as a survival strategy. In habitats where food availability is unpredictable, being able to simply shut down and wait without starving gives tardigrades a decisive advantage over competitors that cannot tolerate fasting. When water returns and the ton reactivates, the tardigrade must feed promptly to replenish its reserves and prepare for the possibility of another dormancy cycle. This alternation between feasting and fasting, between activity and stillness, is the rhythm of tardigrade life, a pattern repeated endlessly across the mosses and lichens of our world. Some species can detect and choose preferred food sources. Despite their simple nervous systems, tardigrades are not indiscriminate feeders. Research has shown that some species display clear preferences for certain food types, seeking out particular algal species, moss cells, or bacterial strains over others available in the same habitat. This selectivity suggests that tardigrades possess sensory abilities, likely chemical, that allow them to detect and distinguish between different food sources. When presented with a choice, some species will consistently move towards one type of cell and ignore another, indicating that their feeding behavior is guided by more than simple chance. The mechanisms behind this food preference are not yet fully understood, but they may involve chemoreceptors on the tardigrade's body surface or around its mouth region that detect molecules released by potential food items. This ability to choose food is ecologically important because it means tardigrades are not just passive consumers, but active selectors within their microhabitat, shaping the composition of the microbial and plant communities around them. It is a small detail, that reveals a greater sophistication in tardigrade behavior than their simple anatomy might suggest. Researchers use them to probe the boundaries of what life can endure. Tardigrades have become one of the most important model organisms for studying the absolute limits of biological survival. By subjecting them to increasingly extreme conditions in the laboratory, researchers are mapping the boundaries of what life can withstand, and tardigrades consistently push those boundaries further than any other multicellular animal. They have been exposed to temperatures from near absolute zero to above 150 degrees Celsius, pressures up to several thousand atmospheres, radiation doses hundreds of times the lethal limit for humans, and prolonged vacuum conditions. Each experiment adds a new data point to our understanding of what is biologically possible. The results inform fields as diverse as astrobiology, medicine, materials science, and conservation biology. By understanding how tardigrades protect their cells, scientists hope to develop new strategies for preserving biological materials, from vaccines to organs for transplant, under conditions that would normally destroy them. Tardigrades have become a living benchmark for resilience, the standard against which all other extremophile organisms are measured. Their willingness to survive almost anything we throw at them makes them invaluable partners in the ongoing effort to understand the full potential of life. Their molecules inspire new ways to preserve vaccines without refrigeration. One of the most promising practical applications of tardigrade research is in the field of vaccine preservation. Many vaccines require continuous refrigeration from the moment of manufacture to the point of injection, a requirement known as the cold chain. In parts of the world where reliable refrigeration is scarce, this cold chain frequently breaks down, rendering vaccines ineffective and costing lives. Tardigrade proteins, particularly the intrinsically disordered proteins that form a protective glass when cells dry out, have inspired scientists to develop new methods of preserving biological materials in a dried, shelf-stable form. By mimicking the tardigrade's vitrification strategy, researchers have shown that certain vaccines and sensitive biological molecules can be dried and stored at room temperature for extended periods without losing their potency. When water is added, the dried material rehydrates and becomes functional again, just like a reviving tardigrade. This approach could revolutionize vaccine distribution in developing countries, eliminating the need for refrigeration and making life-saving treatments accessible to millions more people. It is a powerful example of how the quiet biology of a microscopic creature can have a profound impact on human health. Electronics engineers study their proteins for radiation hardening. Beyond medicine and biology, tardigrade research has attracted attention from the electronics industry. Engineers developing radiation-hardened electronics for use in space, nuclear facilities, and other high-radiation environments are studying how tardigrade proteins provide protection against ionizing radiation. The DSUP protein, which shields tardigrade DNA from radiation damage, offers a biological model for the kind of protective mechanisms that engineers would like to replicate in electronic components. While electronic circuits and biological cells are very different systems, the underlying challenge is similar: how to protect sensitive structures from damage caused by high-energy particles. By understanding how DSUP and other tardigrade proteins absorb or deflect radiation at the molecular level, materials scientists hope to develop new coatings or materials that could extend the lifespan of electronic components in radiation-heavy environments. This cross-disciplinary approach, borrowing solutions from biology to solve engineering problems, is part of a broader movement known as biomimicry. The tardigrade, An animal that has spent 500 million years perfecting its defenses may hold the key to making our technology more resilient as well. Human hibernation research looks to their dormancy mechanisms. The idea of placing humans in a state of suspended animation for long-duration space travel or medical emergencies has been a dream of science for decades. Tardigrades, with their proven ability to enter deep dormancy and revive without apparent harm, are now informing that dream. Researchers studying human hibernation and therapeutic hypothermia are examining the molecular mechanisms that allow tardigrade cells to shut down, survive extended periods without metabolism, and restart cleanly. The vitrification process, in which tardigrade cells replace water with protective proteins and sugars to form a stable glass, is of particular interest. If similar processes could be adapted for human tissues, it might one day be possible to preserve organs for transplant for much longer than current methods allow, or to place patients in a protected state during medical emergencies. The challenges are immense, as human cells are far larger and more complex than tardigrade cells. But the tardigrade provides a proof of concept that biological dormancy at the cellular level is achievable. Each new insight into how tardigrades manage their remarkable transitions brings the prospect of human hibernation a small step closer to reality. Their DNA shielding protein has been successfully expressed in human cells. The successful expression of the tardigrade DSUP protein in human cells remains one of the most striking examples of cross-species genetic engineering in recent years. In the original experiment, researchers isolated the gene encoding DSUP from the genome of a tardigrade species and inserted it into cultured human kidney cells. The modified cells produced the DSUP protein, which then bound to the human DNA and provided a measurable degree of protection against X-ray radiation. This was not a theoretical prediction, but an observed, quantifiable result, with the DSUP-expressing cells suffering roughly half the DNA damage of control cells exposed to the same radiation dose. The experiment demonstrated that a protein evolved in a microscopic invertebrate over hundreds of millions of years could function correctly in a human cellular context. It raised the tantalizing possibility that tardigrade-derived proteins could one day be engineered into human cells to provide enhanced protection against radiation, whether for astronauts, nuclear workers, or cancer patients undergoing radiotherapy. While clinical applications remain far in the future, the fundamental proof of concept has been established, and the DSUP story continues to unfold. Insights from tardigrades may inform future radiation therapies. The understanding of how tardigrades protect themselves from radiation has the potential to influence the future of cancer treatment. Radiation therapy is one of the most effective tools in oncology, but it comes with significant side effects because the radiation that destroys cancer cells also damages healthy tissue. If the protective mechanisms of tardigrades, particularly the DSUP protein and their efficient DNA repair pathways, could be harnessed or mimicked to shield healthy cells during treatment, the side effects of radiotherapy could be dramatically reduced. Researchers are exploring several approaches, from developing drugs that temporarily activate protective pathways similar to those used by tardigrades, to engineering targeted delivery of protective proteins to healthy tissues before radiation exposure. The work is still in its early stages, but the principle is sound. Nature has already solved the problem of surviving extreme radiation, and tardigrades are the proof. By studying their biology in detail, scientists hope to translate millions of years of evolutionary refinement into therapies that could improve the lives of cancer patients around the world. The tiny water bear may yet leave a mark on medicine far larger than its microscopic frame. Bioengineers study them for long-duration spaceflight survival. As space agencies around the world plan missions to Mars and beyond, the challenge of keeping astronauts healthy during years-long journeys has become a central concern. Tardigrade biology is now part of the conversation. Bioengineers studying long-duration spaceflight survival are examining how tardigrade proteins and dormancy mechanisms might inform the design of protective systems for human crews. The radiation environment beyond Earth's magnetosphere is one of the greatest threats to astronaut health, and tardigrade-derived solutions for radiation shielding are being actively investigated. Beyond radiation, researchers are also interested in how tardigrades tolerate desiccation, temperature extremes, and metabolic shutdown, all of which have potential analogies in the challenges of space travel. Could tardigrade-inspired preservation techniques help store blood, tissues, or even whole organs for use during emergencies on a Mars mission? Could protective proteins be incorporated into the materials used to build spacecraft or spacesuits? These questions are driving a new wave of tardigrade research with practical spacefaring applications. The microscopic water bear, it seems, may play a quiet but important role in humanity's journey to the stars. They challenge our definitions of what being alive truly means. Tardigrades in their ton state occupy a philosophical gray zone that challenges our fundamental definitions of life. In that dormant form, they show no metabolism, no movement, no growth, no reproduction, and no detectable response to stimuli, all of which are traditionally listed as the defining characteristics of living organisms. By these criteria, a tardigrade ton is not alive. Yet it is not dead either, because it retains the complete capacity to resume all of those functions the moment water returns. This in-between state has prompted biologists and philosophers alike to reconsider what it truly means to be alive. Is life defined by what an organism is doing, or by what it has the potential to do? If a tardigrade can pause all signs of life for decades and then restart without missing a beat, where exactly does living end and non-living begin? These questions have no easy answers, and tardigrades force us to confront the limitations of our categories. They suggest that life is not a binary switch, but a spectrum, with active metabolism at one end and dormant potential at the other, and tardigrades sitting quietly somewhere in between. In a ton, their body water can drop below 3%. The extent to which tardigrades can dehydrate during dormancy is almost beyond belief. In the ton state, their body water content can drop below 3%, compared to the roughly 80% water content of most active animals. At such extreme levels of desiccation, there is virtually no free water left in the body at all. Every drop has been expelled or locked into the glassy matrix of protective proteins and sugars that stabilize the cells. For comparison, most organisms begin to suffer irreversible cellular damage when they lose just 15 to 20% of their body water. A tardigrade losing over 97% of its water and surviving is an achievement without parallel in the animal kingdom. This extreme desiccation tolerance is the foundation of nearly all of the tardigrade's other survival abilities, because it is the dried, glassy state that provides resistance to heat, cold, pressure, radiation, and vacuum. Without it, the tardigrade would be just another tiny invertebrate, vulnerable to the same threats that limit all other organisms. The 3% figure is a quiet number with enormous consequences, the threshold at which life, as we know it, pauses and biology becomes chemistry, waiting for the return of water. Ton metabolism can fall to almost undetectable levels. When scientists attempt to measure the metabolic activity of a tardigrade in its ton state, they often find that it is so low as to be almost indistinguishable from zero. The chemical reactions that drive life, the conversion of food to energy, the repair of cellular structures, the synthesis of new molecules, all effectively cease. Calorimetric measurements, which detect the heat produced by metabolic activity, show no significant output from a dormant ton. Gas exchange measurements, which track the consumption of oxygen and the release of carbon dioxide, similarly show no detectable activity. This near-total metabolic shutdown is the key to the tardigrade's extraordinary endurance. With no metabolism running, there is no consumption of energy reserves, no accumulation of waste products, and no wear and tear on cellular machinery. The tardigrade simply exists in a state of molecular stillness, its biology preserved but not functioning. This is fundamentally different from hibernation in larger animals, where metabolism is merely reduced. In a tardigrade ton, metabolism does not slow down, it stops. And when it restarts, it does so with a completeness and speed that continues to astonish researchers. They were first described in 1773 by Johann August Ephraim Gose. The scientific study of tardigrades began in 1773, when a German pastor and naturalist named Johann August Ephraim Gose first observed and described these tiny creatures through an early microscope. Gose was struck by their appearance and behavior, noting their plump bodies and slow, deliberate gait as they moved through samples of moss and lichen. He was charmed enough to give them the affectionate name Kleiner Wasserbeer, meaning little water bear, a name that has endured in popular usage ever since. Gose's description was one of the earliest scientific accounts of a tardigrade, and it opened a new chapter in the study of microscopic life. 3 years later, the Italian biologist Lazzaro Spallanzani gave them the formal name tardigrada, meaning slow steppers, which became the basis for their scientific classification. These early naturalists could never have imagined the full extent of tardigrade resilience, as the sophisticated experiments that would reveal their survival abilities lay centuries in the future. But Gose's first careful observations laid the foundation for everything that followed, a quiet beginning to what would become one of the most remarkable stories in all of biology. The name tardigrade means slow stepper in Latin. The formal scientific name tardigrada is derived from Latin and translates directly as slow steppers. It was coined by the Italian naturalist Lazzaro Spallanzani in 1776, just 3 years after Gose's initial description. Spallanzani chose the name after watching these tiny creatures move under his microscope, impressed by the unhurried, deliberate way in which they placed each of their 8 stubby legs. The name proved to be brilliantly apt, capturing in 2 Latin words the most immediately recognizable behavior of these animals. It has remained the official scientific designation for the entire phylum ever since, spanning over 2 centuries of classification and study. The choice of a name based on movement rather than appearance was an insightful one, because it highlights the behavior that most distinguishes tardigrades from other microscopic organisms. While many tiny creatures dart, swim, or wriggle through their environments, tardigrades walk slowly and carefully as though they have all the time in the world. Given that their lineage stretches back over 500 million years, perhaps they do. The name slow stepper is not just a description, but a philosophy of existence, one that has served tardigrades remarkably well. The term perfectly reflects their careful, lumbering gait. The name slow stepper is not merely a convenient label. It is a precise description of one of the most distinctive features of tardigrade behavior. Under the microscope, a tardigrade in motion looks remarkably like a tiny bear making its way across an uneven landscape. Each leg moves in a careful, coordinated sequence, gripping the substrate with its claws before the next step is taken. There is no rushing, no darting, and no sudden changes of direction. The gait is steady and purposeful, a rolling motion that propels the plump body forward with a kind of quiet determination. Scientists studying tardigrade locomotion have found that their walking patterns share similarities with those of much larger arthropods, suggesting that these basic movement strategies are deeply conserved across evolutionary time. The slowness of their gait is not a limitation, but an adaptation. In the tangled three-dimensional world of moss fibers and soil particles, speed is less important than sure-footedness. A careful step is worth more than a fast one when your world is made of slippery surfaces and precarious footholds. The name tardigrade, the slow stepper, is a tribute to this quiet wisdom, an acknowledgement that patience and care are virtues even in the smallest of creatures. More than 1,300 species have been identified so far. As of recent counts, scientists have formally described more than 1,300 species of tardigrades, distributed across 3 main classes and numerous families. This diversity spans an impressive range of habitats, body sizes, feeding strategies, and reproductive modes. Some species are tiny, barely 1/10 of a millimeter long, while others reach 1/2 millimeter, giants by tardigrade standards. Some are herbivores, some are carnivores, and some are generalists that exploit whatever food is available. The classification of tardigrades relies on detailed examination of features such as claw shape, pharyngeal structure, cuticle ornamentation, and egg morphology, all observed under high-powered microscopes. Each new species description adds to our understanding of the evolutionary breadth and ecological versatility of this ancient phylum. The current count of over 1,300 species is almost certainly an underestimate, as many habitats have yet to be thoroughly surveyed, and the microscopic size of tardigrades means they are easily overlooked. With every new field expedition and every moss sample examined, the known diversity of tardigrades continues to grow, revealing ever more of the variety hidden within this remarkable group. New species are still being discovered regularly. The pace of tardigrade species discovery shows no signs of slowing down. Each year, researchers around the world describe new species from habitats ranging from Antarctic mosses to tropical tree canopies to deep ocean sediments. The microscopic size of tardigrades, combined with the vast number of unexplored microhabitats on our planet, means that the catalog of known species is likely only a fraction of the true diversity. Some estimates suggest that the total number of tardigrade species could be several times higher than the current count of just over 1,300. New molecular tools, including DNA barcoding and environmental DNA analysis, are accelerating the rate of discovery by allowing researchers to detect and distinguish species that look identical under the microscope but differ genetically. Citizen science projects, in which amateur microscopists collect and examine moss samples from their local areas, have also contributed to the discovery of new species. Every new addition to the tardigrade family tree deepens our understanding of how these creatures have diversified across the planet and adapted to such a wide range of environments. The story of tardigrade taxonomy is still being written, one tiny species at a time. Some species are named after Star Trek characters. In a delightful tradition that reveals the playful side of taxonomy, some tardigrade species have been named after characters from popular culture, including figures from the Star Trek franchise. The connection between tardigrades and science fiction was strengthened when the television series Star Trek Discovery featured a giant tardigrade as a key character, capable of navigating through a network of cosmic spores. This fictional portrayal, while scientifically fanciful, brought real tardigrades to the attention of millions of viewers. In tribute, some researchers have chosen to honor the connection by naming newly discovered species after Star Trek characters and terminology. This practice is part of a broader tradition in taxonomy where scientists name new species after people, places, or cultural references that hold personal significance. It adds a touch of warmth and personality to the otherwise technical business of classification. For tardigrades, whose real-life abilities are arguably more impressive than any fictional version, having species named after science fiction characters feels fitting. These tiny creatures have already crossed the boundary between science and popular imagination, and their Star Trek namesakes reflect that journey. Others honor the scientists who first collected them. Many tardigrade species bear names that honor the scientists, naturalists, and collectors who first found and described them. This is one of the oldest and most respected traditions in biological taxonomy, a way of acknowledging the individuals who dedicated their time, skill, and curiosity to the study of the natural world. The names often take the form of Latinized versions of the person's surname, appended with a species suffix. For researchers who have spent years studying tardigrades, having a species named in their honor is one of the highest forms of recognition within the field. Some of these commemorated scientists were pioneers of tardigrade research, spending decades collecting moss samples and painstakingly examining them under microscopes to identify new species. Others were general naturalists or explorers who happened to collect samples that later yielded new tardigrade discoveries. The tradition ensures that the contributions of these individuals are preserved in the permanent record of biology, their names attached to the very creatures they help to reveal. It is a quiet form of immortality, a legacy written into the scientific literature and carried forward by every future reference to the species they described. They have become cultural icons of resilience and survival. In recent years, tardigrades have transcended the boundaries of scientific research and become cultural icons, symbols of resilience and survival that resonate far beyond the laboratory. Their image appears on merchandise, in documentaries, in educational materials, and across social media, where they are celebrated as the toughest creatures on Earth. The appeal is easy to understand. In a world that often feels uncertain, the idea of a tiny animal that can survive anything, from the vacuum of space to 500 million years of geological upheaval, carries a powerful emotional resonance. Tardigrades represent the idea that toughness does not require size, that patience can outlast destruction, and that quiet endurance is a form of strength. Schools use tardigrades to teach children about biology and resilience, and motivational speakers cite them as examples of perseverance. Scientists, for their part, are generally delighted by the public's affection for these creatures, as it brings attention and funding to a field that has long operated in relative obscurity. The tardigrade has become a tiny ambassador for the natural world, carrying a message of hope and endurance wrapped in a body smaller than a grain of sand. They appear in cartoons, toys, and internet memes. The cultural popularity of tardigrades has extended well beyond the scientific community, with these tiny creatures now appearing in cartoons, children's toys, plush dolls, video games, and countless internet memes. Their distinctive appearance, plump, roundish, and oddly endearing, lends itself perfectly to cartoon representation, and artists have embraced them with enthusiasm. Tardigrade plush toys have become popular gifts among science enthusiasts, and their image adorns everything from mugs and t-shirts to phone cases and stickers. On the internet, tardigrade memes celebrate their toughness with humor, often depicting them as indestructible superheroes or comparing their resilience to everyday human struggles. This pop culture embrace has done more to raise awareness of tardigrades than decades of scientific publication could have achieved. It has introduced millions of people to the idea that the microscopic world is full of wonders and that the tiniest creatures can be the most fascinating. For a phylum that spent most of its public existence in obscurity, the transformation into a cultural phenomenon is remarkable. The water bear has become a star, quietly conquering the internet with the same patience it has shown for 500 million years. To scientists, they symbolize life's astonishing persistence against all odds. For the researchers who study them, tardigrades are more than just laboratory subjects. They are living symbols of life's astonishing capacity to persist against all odds. In a universe that is overwhelmingly hostile to biology, where the vacuum of space, the cold of deep ice, and the violence of radiation represent conditions that should be incompatible with anything alive, tardigrades quietly demonstrate that life can find a way. Their survival strategies, refined over more than 500 million years, represent one of evolution's most elegant solutions to the problem of existence in an unpredictable world. Every tardigrade that revives from a dried ton, every individual that walks away from a radiation dose that would kill 1,000 humans, is a testament to the resilience that is woven into the fabric of life itself. Scientists study tardigrades not just to advance knowledge, but to be reminded of what biology is capable of when pushed to its limits. As you rest tonight, consider the tiny water bear, somewhere in the moss and soil of our world, curled patiently in its ton, waiting for the rain that will call it back to life. It has been waiting for a very long time, and it will go on waiting, quiet and resilient, for a very long time to come. And now, the quiet wonders of the tardigrade world begin to gently fade into the stillness of the night. You have wandered through over 100 peaceful truths about these microscopic survivors, from their ancient origins in prehistoric seas to their accidental journey to the moon, from their ability to endure the vacuum of space to their patient wait through decades of drought, from the strange proteins that shield their DNA to the simple elegance of their transparent bodies. You have explored how tardigrades dry into tons and pause life itself, how they repair damage that would destroy other organisms, and how they thrive in places from mountaintops to ocean depths, from tropical forests to Antarctic ice. You have learned that these tiny creatures, smaller than a grain of sand, have outlasted the dinosaurs, survived 5 mass extinctions, and continue their slow walk across the moss and lichen of our world. You are connected to this story more deeply than you may realize. Right now, as you listen, tardigrades may be resting in the moss on your roof, the soil in your garden, or the lichen on a nearby tree. They share our planet with quiet determination, asking only for a thin film of water and a place to walk. If your eyes feel heavy now, let them close. Let the rhythm of your breath become steady, like the slow, deliberate steps of a water bear crossing a moss leaf, like the patient pause of a ton waiting for rain. Tonight, as you rest, tardigrades continue their ancient existence all around you. They curl into dormancy and wait out the night. They carry on their quiet lives in places we rarely notice, enduring whatever comes with a resilience that spans geological ages. You are a warm presence within a world filled with such quiet wonders, peacefully resting among the microscopic marvels. Sleep well, sweet dreams, and may you carry with you the quiet knowledge that resilience is not always about strength or size, but about patience, adaptation, and the stubborn will to endure. That somewhere in the moss and soil of our world, tiny water bears are walking slowly through the night, survivors of 500 million years, unbothered by the vastness of time. Thank you for joining me on this journey through the remarkable world of tardigrades. If you enjoyed this video, please subscribe to Drowsy Discovery for more soothing explorations of the wonders hidden in our world. There are more wonders waiting, more secrets hidden in the microscopic realms we rarely see, more gentle truths to drift through on quiet evenings. Rest well, and may the quiet persistence of the tardigrade keep you company through the night. This has been Drowsy Discovery. Good night.